Good morning, everyone. Welcome to day 2 of the GIAN course. Hope everyone had a good night's rest. I did, so yeah. So yesterday we covered a few concepts on radiology informatics, where we covered basics of radiomic feature extraction and how to use some of the more advanced concepts, more context-driven approaches for radiology AI, including human in the loop or radiologist in the loop AI systems. So if you have any questions regarding that, we can always discuss after this lecture. But today I want to switch gears and talk about AI in digital pathology. So how many of you here are, let's say, familiar to some extent with what digital pathology is? I know that I briefly introduced it yesterday, but any of you here have worked with digital pathology? So what comes to your mind when you hear the words digital pathology? Anyone?
Glucometer, sir. Glucometer pathology. We are testing the sugar test in the morning. So that is digital
no,
pathology. I'm thinking.
It's not that.
Yes.
So let's have the mic here.
Sir, I think using AI to detect some kind of disease pathologically.
So I think that's the general theme of this entire series, right? So we are using AI to do something to detect disease and prognosis and whatnot. But what comes to mind when you hear about digital pathology? So maybe we can have the mic here. Yeah.
So in traditional we have this slide mechanism where you have slide, you scan it, but in
Right.
digital we have a whole slide imaging, so that we take the scan of entire slides and later process using some techniques and diagnose and pre-process those things.
Right. So what happens traditionally is, so if there's some, let's say, suspected nodule or suspected lesion, what happens is the surgeon would go in, resect or take a biopsy of that particular region, and then they would take it out, mount it on a glass slide. They would fix it with different dyes, and then they would give it to a pathologist, and the pathologist can then review that slide under the microscope. So why does a pathologist do that? So if the pathologist does that, they can then look for any abnormalities, if there are any cancer cells or some other regions of interest that can point to some disease, right? So that's the traditional mechanism of how pathology works. Now in digital pathology, what happens is you take that slide and you digitize it, and then instead of the pathologist having to look at it under the microscope, they would look at the computer screen, look at the slide, look at the image, and do whatever analysis they have to do later on. So in the course of the next hour and a half, what we will do is we will cover some of the fundamentals of digital pathology, especially computational pathology. What are the advantages, how these can be used, and how in fact this is sort of a paradigm shift in the domain of pathology. So what is digital pathology? So like I said, it incorporates the acquisition, management, sharing, and interpretation of digitized slides. So all of these terms, acquisition, management, sharing, and interpretation, all of these are important because by combining all of these, we have essentially moved from a more traditional analysis of these pathology images to more modernized analysis, where we look at these images, or the pathologists look at these images on the screen. So starting from the process of acquisition, so how these are digitized, to management, because these are images, right, and these are huge images, so you need a lot of storage. And then sharing, so because these are images, you can share them, and then share them. So traditionally what happens is, if you have a slide, you would ship the slides, right? You would physically ship the slides. So there's no more shipping of the slides, no more couriering the slides. So you can actually send it via internet. And then interpretation. So this multiple pathologists can sit at the same time and they can interpret the same image at different locations. So digital slides are created when glass slides are captured with a scanning device, and we'll see what the scanning device looks like. what this does is it provides a high resolution digital image that can be viewed on a screen or a mobile device. So imagine, let's say a pathologist is traveling, right? So they're not at the desk, so they don't really have access to a microscope. But if the slide can be digitized and it can be transferred to their, whatever server there is, they can, if there's a proper viewer, they can open it on their phone, on the smartphone, and can view it. So it enables something called telepathology. So you can capture the entire glass slide, and so we heard a couple minutes ago the term whole slide image. So that's when you capture the entire glass slide under bright field or fluorescent conditions at a magnification that is comparable to what you can actually observe under a microscope. And last but not the least, you can actually share it using specialized pathology software applications. So now the buck doesn't really stop there. Yes, this process has been digitized, but then where can we come in? As imaging researchers, as scientists, where do we come in? So now that we have access to these digital images, we can apply our fancy AI tools, our mathematical models, and analyze these images in a way that's almost impossible by just looking at these images, similar to what we saw in radiology yesterday. So here's a summary. You have the traditional pathology workflow here, where you have the tissue preparation, and like I said, if you don't really have an expert in that physical location, you have to ship the slides to a lab where it is analyzed, and then you have a patient diagnosis. However, with digital pathology, after the tissue preparation, you have the step which creates the digital slides, and then everything is computerized after that. So you have the analysis of the digital slides. I mean, obviously you have the pathologist who can look at the digital slides, but then you can build your AI systems for automatically diagnosing diseases or doing whatever, let's say, a morphometric analysis that you have to do. So one of the first benefits of digital pathology is quality. So there are algorithms for analyzing slides, and these are objective, accurate, and quicker than microscopy. So imagine a pathologist has to look at a slide under the microscope, they would take some time. But if you can actually digitize it, so instead of spending time on getting the slide, mounting it on your microscope, and trying to move it, everything can be done with the click of a mouse, right? So, and there's rapid access to prior cases. When it comes to looking at a lot of times pathologists might be interested in looking at previous slides. If they want to look at previous slides, what they have to do is they have to, let's say, unmount the current slide, find out where in the rack the previous slide is, mount it, and then view it, right? So there is some time involved there. I'm not saying that it's a lot of time, but there's some time involved there, right? And these slides have to be stored. I mean, they have to be physically stored. When it comes to digital pathology, you basically store these slides digitally. So you can, at any point, if you know your folder structure, you can go back, you can access your previous slides, and you can look at them and you can analyze them. And the data storage allows for long-term predictive analysis, right? So a lot of times, let's say if you don't have enough physical storage space, or for some reason, the slides that have been there for 10 years, 15 years, there's a chance that the physical slides might get lost or might get damaged. But if you have them digitally, and if you have a backup of that on some server, you will have them theoretically forever, right? So let's say that you want to teach the pathologist wants to teach their residents or their fellows, their medical students. There are some interesting cases that they have observed 5 years ago, 10 years ago. They can keep a note of that on their server, and they can immediately pull that slide, as opposed to having it physically where they have to store it somewhere, right? So that's how it becomes important, and the quality is also enhanced. Errors are reduced. So you have barcoding. So each of the slides has a separate barcode, and based on that barcode, you can pull whatever slide you want, and better views. So you can have zoom in and zoom out at multiple angles, and then you can measure multiple areas of interest. So if one area is important, they can go to that area, immediately zoom in, and they can on their software, they can mark that a particular area. So let's say that you have this whole slide image. last slide, there's this whole slide image, and they're looking at it on a computer screen. So this region is important. They somehow they have some sort of marker and say that this is an important region, and that it's a digitized marker, right? It's just a pen mark, so it stays there, and when they come back, they can always look at it, find out what it is that they annotated, and that can also be used for downstream analysis. Okay, so here's just a summary of what the text here says. You have improved analysis, better views, and it can reduce errors. So all of this is connected to the quality of the images that digital pathology helps us, or digitized pathology slides help us achieve. Now productivity, right? So this is also very important, because one of the main let's say driving factors of research is collaboration. So eventually we would want to collaborate with scientists from other fields. As a pathologist, they would want to collaborate with computer scientists or biomedical researchers to analyze these slides, right? So how do we share the slides? You can't expect to give this slide to a biomedical researcher. They look at it under the microscope, and there's not much that they can do. I mean, maybe they can do some qualitative analysis, but there's not much you can do beyond the qualitative analysis by looking at the slides under a microscope. But if the slides are digitized, you can share it with other people. We routinely have collaborators sharing slides with us. So we can take these images, we can look at it in multiple different ways, and then we can apply our deep learning techniques, our handcrafted feature analysis, and get information which otherwise would be impossible. So it allows for automation and work schedules, remote access. So let's say that, like I said, a pathologist might be traveling, they would want to access these slides remotely. So you can actually do that if you have these digitized slides. So reduced turnaround times, similar to what I discussed in the previous slide. It reduces because of digitization. It reduces time retrieving and data matching, organizing these slides, and speeds up access to samples and improves turnaround time. So why is this important? Because pathologists every day, they have to look at hundreds of slides, right? So just taking slides that are physically there, mounting it on the microscope, reading the slide, analyzing the slide, writing the reports, then putting it back, getting another slide. I mean, yes, traditionally pathologists are used to that, but imagine if something like this can be done digitally, it would definitely improve the turnaround time. But obviously, that doesn't mean that this change would happen overnight. So there has been a steep learning curve that has been observed when it came to digitizing these slides, and not just digitizing the slides, going from a traditional pathology workflow to a digitized workflow. So there are traditional pathologists who still resist it, because they're used to their previous protocols. Now there will obviously be a change in protocol, but everyone has to adapt, and slowly they have been adapting, and there are multiple hospitals over the world that have gone completely digital when it comes to pathology. And where it becomes very important for us is innovation. So eventually this is big data, right? So there are, if we can digitize these slides, there's so much data, so much information that we can obtain from these images. So it allows pathologists to become more specialized. So let's say that a pathologist wants to completely focus on prostate cancer, or completely focus on breast cancer. So now they have, so with digitization, they can actually spend more time on a particular field. They can spend more time doing research in that particular field, right? So it can also help us deliver better tools for teaching and training. So imagine there's a microscope, and I'm sure most of you here would have been exposed to some microscopes in your high school or in your college, junior college. So imagine that your teacher, your professor wants to show you something under the microscope. So they would adjust the slide, and they would ask you to come there and take a look and see, okay, you see this particular portion of the slide, this is what it means. Now, so just for a second, imagine if there were 10 students in the class, right? So the teacher would, or your professor would identify that area of interest, and one by one they would ask each of you to come and observe. And this is what happened when I was in +2, and then I was doing some, I think they were looking at some, I don't know, it was some seed or something that in our botany class, I remember, right? So now if it was digitized, let's say you take that slide, you digitize it, and now instead of having each student come one after the other and then look at the slides, you can actually display that slide, right? You can display that slide, you can have everyone look at the same slide. So this is how it can actually help in teaching and training. So even if, for a second you say that maybe let's not use it for diagnosis, we can still use a traditional method, that's fine, but you can use these techniques for teaching, right? So that's why it can improve productivity. And a summary of, how it can improve workflows, there's more innovations, and reduced turnaround times. So all of it results in increase in productivity of pathologists. Okay, so what is done? So you have a scanner, a whole slide scanner. You scan a complete microscope slide and create a single high resolution digital slide. And we'll see in a few minutes what this high resolution means. So you can capture many high resolution image tiles or strips, and then you can montage them to create a full image of a histological section. So the goal is to achieve efficient and cheaper diagnosis and prognosis and prediction of diseases. So yesterday we saw what diagnosis means, what prognosis means, and what treatment response prediction means. And all of this has been made possible by AI and the different machine learning tools that we are developing. So what are the benefits for patients? Let's say routine screening. It can help in enabling proper quality and safety of patient care, and increased efficiency and standardization. So what do I mean by standardization? So what I mean by that is, so let's say that you have, at site 1, you have your traditional ways of pathology analysis, and site 2 you have traditional way of pathology image analysis. A lot of times it becomes very subjective, right? So this results in some sort of loss in standardization. But now let's say that you have these slides that you can send to a center where it can be digitized, and then it can come back. So there's some level of standardization because the slides have been standardized under the same let's say digitization protocol, right? So there's some level of standardization that can be brought about by digital pathology. This is how a slide scanner looks like, right? So what happens is you have, let's say you have multiple different glass slides. The glass slides go in here, so you have the slots here, and once they go in here, they can be scanned. You have a high-powered camera here. They are scanned, and then you see on your display how the image looks like, as opposed to having a microscope and putting that under the microscope and then looking at it. And there are multiple different companies that do this, Hamamatsu, Roche, and there are multiple different companies that can do this. Okay, so I hope I can play this video.
The number of patients in the UK that have to wait over 6 weeks for a diagnosis has increased 17% annually since 2010. This means that people like you and me have to wait much longer to find out if we have cancer or not. But why does the process take so long? Let's look at the current method. The process begins with a doctor taking a tissue biopsy from the patient. This sample is then sent to a pathology lab where it begins its preparation stage. The tissue is cut into thin sections, stained, and put onto glass slides. The sample is then referred to a histopathologist. That's someone who specializes in diagnosing diseases. They examine the sample and make a diagnosis. If they're unsure, the sample is packaged and sent to another histopathologist for a second opinion. The package could be sent to another town, another city, or even out of the country. This increases the waiting time by days or even weeks. This can result in unnecessary pressure and stress being added onto patients. Surely, with all the technology we have today, there must be a faster way of diagnosis. Luckily for us, there is a technology that could make a difference. Digital pathology. If we go back to where we started, Instead of the tissue sample being packaged and sent away, the sample is digitally scanned and uploaded onto a secure server, where only verified pathologists can access it. The easy accessibility means that other pathologists can come together and collaborate in giving their opinions and suggestions. The pathologists can also examine the sample in great detail and make notes and highlight areas, aiding the communication between pathologists. Going digital can significantly reduce the diagnosis time. This means that patients can get their results much quicker than traditional methods and can begin treatment much earlier if necessary. So now, let's talk about the possibilities that digital pathology can support. With the current advancements of computing technology, pathologists could soon utilize the power of artificial intelligence, helping them to achieve a faster, more precise diagnosis. Artificial intelligence could help assist with identifying abnormal tissue or cancer, with quantifying the severity of disease, and it could help support a more personalized approach to treatment. This streamlined approach will improve diagnostic workflow for pathologists, improved support services for patients, better task prioritization, and helping assist pathologists in diagnosis. Why do we need digital pathology to be widely used across the NHS? In England alone, 13 million histopathology slides are examined each year. Since 2007, there has been a rising demand for diagnostic tests. This has coincided with a reduced pathology workforce capacity. A recent survey by the Royal College of Pathologists discovered that only 3% of pathology departments had enough staff to deal with this increasing demand. For the NHS, digital pathology will help meet these challenges and will provide patients with faster results and better care. Digital pathology, the next step in achieving improved diagnostic services and results for patients.
Okay, so this is
The number of
basically the summary of what I covered in the past few slides, and digital pathology is completely reshaping the way pathologists analyze images. So I was there at Tata Memorial a couple weeks ago. So they have an entire department that is dedicated to digital pathology. They have, I think, about 5 to 6 scanners, and all the slides that they get is digitized, and currently their pathologists are analyzing everything in a digital manner. So it will take time for other hospitals to catch up, but eventually, definitely in the next couple decades or so, the future will be completely digital. Okay, so this is not something new. This is not something that started 5 years ago or 10 years ago. The foundations of this were laid almost 50 to 60 years ago. And so there's an important lesson here, because we might think that a lot of these techniques that we are developing nowadays, yes, you write a paper and you forget about it, and you think that, okay, this probably will never see the light of the day. But that's not true. If you're diligent in doing whatever you're doing, and there's a significant unmet clinical need that you are addressing, eventually if there is need for that technique, it will show up in mainstream in the next 20, 30 years or so. Back in the day, I don't think no one even believed that technique like digital pathology could take off. But now you see how it's completely changing. The scene has completely changed. And obviously it's not, it did not happen just by itself. A lot of it was helped by the AI boom, right? You have a lot of compute infrastructure now, how you have different GPU servers, and processing of these images has become a lot faster. So all of that has aided digital pathology, right? So it all comes down to the fact that it's a collaborative thing, right? You can't just stand up a digital pathology unit by itself. You need the buy-in of the hospital IT, so that they can invest in compute infrastructure. You can have enough GPUs, you can have enough compute power that can support these kinds of activities. You need specialized hardware. It's not just that you take a camera and digitize it, right? You need specialized hardware. So, like I said, the benefits, they include medical education. So, telepathology is important because you might not have, so a lot of times you'll see that, for example, in India, you might not have specialized pathologists. So, so you might not have specialized pathologists at all places, right? So, there will be specialized pathologists somewhere else sitting remotely, but you would want to get their opinion. So, let's say that there's a breast cancer slide that needs to be analyzed, and your hospital only has general pathologists who will be able to provide their interpretation, but in order to get something more specialized, it has to be sent to a specialized pathologist who specializes in breast cancer. Now, in order to do that, you can scan that image, you can digitize it, and you can just send it over to them and get their opinion, right? Or what you can do is you can open screen sharing, and then you can have a collaborative session where the specialized pathologist basically looks at the same region as you are looking, and they would annotate what regions are important, or if there's a particular disease that that tissue has. While they call what disease it has, they can tell you what parts of the image was responsible in detecting that the patient has that disease, right? So, you can see how it can enable collaboration, how it can enable telepathology. So, clinical workflows, it can obviously improve clinical workflows, make it faster, and research. It definitely has played a huge role in developing AI-based biomarkers for many different diseases. So, pathology education. So one of the ways of using digital pathology for education, like I said, is there are, let's say, a group of medical students who are doing a pathology rotation. So the pathologist would want them to understand that, okay, there's a particular image, and certain parts of the image has, let's say, certain diseases. So they can digitize it, put it up on the screen, and show them what the different parts of the image are. So, and it's interactive, right? So they can play around with the image. You can zoom in, you can zoom out, you can basically pan, and you can do all sorts of, you can maybe increase and decrease the brightness, or let's say that you apply some thresholding thresholding techniques to identify where there are particular structures. You can do that. So you can play around with the intensity of the image. So you can do all of these operations on your the software that you have. So if any of you is interested in looking at or playing around with digital pathology slides, you can access the TCGA website. So TCGA is an initiative in the US where they are collecting a lot of images and corresponding genomic data, and for a lot of these data sets that are out there, there's also corresponding radiological data on TCIA, the Cancer Imaging Archive. So this is a very rich resource. So I know that the last couple months there was something going on with the repository that we could not access many of these images, but I think they're probably back. But I'm sure if they're not there, they'll be there in a few months or so. So I think they have a collection. Last I checked, they had a collection of over 11,000 or 12,000 whole slide images, and this spanned multiple different cancers. So they have a very good interface. If you want to, let's say, look at only brain cancer, you can go there, you can build your own cohort that there's a particular cancer type you need. If there's some genomic data you are interested in, you can download that data, and you can also see if there's matched imaging data. So not all the cancers have imaging data. By imaging, I mean radiology data, but there are multiple of these cancers, especially lung cancer, which has, I think, rad path omics for about 150 studies or so. So yeah, I mean, don't quote me on the numbers. They keep changing. People keep adding their data sets. And also, let's say that here you are generating some data sets, you are generating some analysis results. You can upload your results to TCGA, right? You can I mean, obviously there needs to be, it's good if you have a corresponding publication. So one of the avenues that people have taken in the last few years is Nature Scientific Data. So that's a good journal where, let's say, you have some data sets that you have that you want to upload or you want to contribute to the scientific community. You have some analysis results. The data has to have some analysis results with it. Data cannot stand on its own. So you write a scientific data paper, and once it gets accepted, you can tell them, or you can obviously talk to the TCGA folks, and you can upload your analysis results. So let's say that there's some new feature that you've developed, some new biomarker that you've developed, and you want to compute this biomarker for each of the whole slide images or each of your radiology images that you have, and you want the community to use it. So eventually we want to do everything for the community to advance science, right? So if you think that there's merit, or you think that it will benefit researchers like you or medical researchers, by all means you can approach TCGA, TCIA, they'll be very helpful, and you can upload your images, you can upload your omics data and your analysis results. So there is also another resource, I haven't noted it here, but I can write it down, Imaging Data Commons. So Imaging Data Commons is a relatively new initiative. I think it started in the last, it's been less than 5 to 6 years. So what you can do there is you can also upload your scripts. And when you upload your scripts, they do provide some compute. It can directly interface with TCGA or TCIA. You can pull images, run it on their system. Right? So let's say that you have developed a new algorithm for quantification of cells, or let's say you have developed a new algorithm. So this is mostly for radiology, for blood vessel analysis like we saw yesterday. You can always upload your code, you can talk to the IDC folks, they can help you integrate it, and you can pull images that you haven't analyzed yet, and you can run it on their platform. So this is a relatively new initiative, this I think at Harvard, yeah. Okay, so how else can we use digital pathology and AI for teaching, for education? We'll cover some of that tomorrow and day after. We'll cover generative AI tomorrow and day after. I think Dr. Baig also has a tutorial on that. So imagine that there's some kind of cancer for which you do not have enough images. You do not have enough whole slide, you do not have enough tissue samples, so you do not have enough whole slide images. So what we can do is we can use generative AI techniques like GIANs or diffusion models. We can generate these these synthetic images, and then you can show these synthetic images, and obviously these synthetic images have to be blessed by pathologists so that it looks real. I'm sure you're ready with the question. Okay. So these images have to be blessed by pathology. I mean, these images have to look real, right? It's not just generating a synthetic image. It has to show proper characteristics of disease. If there's certain level of tumor infiltrating lymphocytes or certain level of cancerous tissue that has to be seen, then the generated image also has to show that degree of faithfulness. You can't just generate any image and say that because I have a prompt that says generate image of XYZ cancer, that it actually does that. You have to verify that with your pathologist. But once you have verified that with your pathologist, you can actually show that image to a medical student or to a resident or a fellow, and they can understand what these images look like. And also with generative AI, there's another fascinating aspect to it. So let's say that there's a certain disease, and this is a very hypothetical scenario, that for a certain disease there's certain layout, or the structure of the cells appear a certain way, the layout of the cells appear a certain way, the cells appear at a particular position. What you can do is you can actually, let's say you can extract the cells, you can segment the cells, and then you can train an AI model to give you similar images or images with similar cell layouts, but something else that's different, right? So you can essentially extrapolate your images to make it look like something else. So let's say that you have a particular grade of cancer, grade 1, grade 2, grade 3, grade 4. So a lot of times it happens in a continuum. It progresses from grade X to grade Y. So let's say that you have grade X, you have grade Z, and then you can ask your diffusion model or your generative model to interpolate what are the grades in between that, right? So that also helps you in teaching your residents, your fellows, as to how the disease progression happens. So there's the possibilities are almost endless, almost limitless, what you can do with generative AI. Okay. Also, I am not covering this in this lecture, but not all the slides are H&E stains. So H&E is one of the most common type of stains which stains for the nuclei, the background, the epithelia, the stroma. But there are other kinds of stains that are more specialized, that have different kinds of stains for different kind of nuclei. Right? So there are image-to-image translation methods, or there are generative methods where you can take one stain and convert it to another stain. So a lot of times, traditionally how this happens is, you have the tissue block, you section it, and so these are very thin sections. You take the first section, you stain it with one kind of stain, and you digitize it. Then you take the immediately next section, so that they are quite similar to each other. You stain it with another kind of stain. I mean, the stains are what? They're like proteins, right? They would they would attach to different regions of the tissue. So you would stain it and you would digitize it. Now, or another way of doing that is, you take the first stain, after it has been digitized, you take that tissue that has been stained, you wash away the stain, and you restain it with another stain. So you can do that as well. I mean, sometimes it leads to loss in tissue, but that's something that's done routinely. But let's say that you have one stain, and then you lose that slide for some reason, or you don't have the the sections that are around it. So you can take the available digital tissue or digitized tissue, and you can train your generative models to artificially convert it to another stain. That is assuming that you have enough paired data in your training, that you have stain A, stain B, you have enough data for stain A and stain B. You learn a function that can convert stain A to stain B with your training data, and in your testing phase, in your inference phase, you do not have stain B, you only have stain A. Then you can use your model to convert stain A to stain B, right? So that's something that you can do using generative AI. And that's one of the hot areas of research nowadays. How do we artificially stain tissue with different kinds of stains? Okay, so teleconsultation, we covered that. So inter-observer and intra-observer variance. So this is very important, and this holds good not just for pathology, but also for radiology, or for that matter, any kind of image analysis. So because a lot of times what happens is, so today the pathologist would look at an image, and tomorrow they would come back, or let's say after a few days, they would come back, and for some reason they would want to change the diagnosis, or they have something in their mind that would sort of bug them, and say that, okay, I want to go back, relook at my image, and have another look at my image, and see if there's something that I have missed. Now that it's digitized, they would have, let's say, marked the problem areas, and they can immediately pull up the slide on the computer and go back to the problem areas and see if there's something that they missed, and correct that, right? So that's basically intra-observer variance. So inter-observer variance. So you have two pathologists here. Pathologist 1 says something, pathologist 2 says something. Because you have this collaborative setup now with your digitized tissues, they can look at it at the same time, they can sit together, and they can discuss, and then arrive at a consensus if needed, right? So this can help assess this variance, or let's say you are conducting an experiment. So just another hypothetical scenario, you have come up with this really cool generative AI model where you are taking one kind of stain, converting it to another, or you are generating synthetic samples of a particular disease. Now you want the pathologist's opinion as to whether this synthetic sample that you have generated is good, bad, right? Good or bad or ugly or whatever, right? So you have then two different pathologists. You keep them separate, like you keep this study blinded. You provide the same generated image to both the pathologists, and you get their diagnosis, right? Not diagnosis, you get their interpretation as to how real that image is, how real that synthetic image is, or how real that fake image is. So you get their impression, and then you can compare them, right? So digital pathology can help you do that. Archiving of slides, we covered that. So digital reviews of diagnostic workflows. So this is again something very important. So you remember we The last part of yesterday's lecture, we covered about eye gaze analysis, how we can take eye gaze. In fact, Moinak, who's sitting here, he worked on a lot of those projects. So we covered a few projects that we have been doing on eye gaze analysis. Now, what is done in a lot of these digital pathology software is, while the pathologist is looking at different regions of the slide, so I can try to illustrate that here. So this is your computer screen. So let's say you have the whole slide image here. Okay, I lost it. Okay, so you have the whole slide image here. So let's say the pathologist starts. Just a second. The pathologist starts from here, and then they move here, and then they move here, come here. So this is 1, 2, 3, 4. So just for simplicity's sake, let's say that you have these 4 regions that the pathologist traverses while they make their decisions. Now, what a lot of these software do is, they actually keep a record of where the pathologist is focusing their attention on, and they would have something like this that gets saved. This is the whole slide image. They would actually have this path saved. Right? So that is how the pathologist sort of moved around while looking at this image. So this is nothing but connecting the centers of the fields of view, right? So now what this can help you do is, so let's say that, so typically what happens in many of these clinical workflows is that junior radiologists or junior pathologists would make the decisions, they would make the diagnosis, and these have to be reviewed by senior pathologists. Now, if you had a provision like this, where the scan path was saved, it was noted, then if there is any mistake while the senior pathologist is reviewing what the junior pathologist has done, they can tell them that this is the path they followed, or this is what went wrong when they were diagnosing this image. Does that make sense? Yeah. So it can help provide digital, it can help in digital reviews of diagnostic workflows. And through this, you can also provide feedback to pathologists. So remember, for the eye gaze analysis that we saw yesterday, one of the applications that we discussed was teaching, right? So how, if you can train a model to understand the eye gaze of specialists, you can use that to teach, you can in theory use that to teach a junior pathologist or a junior radiologist. So by providing feedback, so if when they're looking at their images, if there's some portion of the image that they're missing or that they haven't really looked at, and if there's something interesting in there, your AI can tell you that, okay, here's an interesting region that you have missed, why don't you shift your gaze, go up here, and then look at that image and see what's going on there. Okay, so for clinical diagnosis, obviously it's important for clinical diagnosis. So it has actually been shown, and this keeps getting better and better. If you compare the diagnosis that's made physically under the microscope and the diagnosis that's made using the digital pathology images, there's a pretty high concordance. And this is only getting better as pathologists are becoming more receptive to the idea of digital pathology. So accuracy of reproduction of glass slide, it increases accuracy, and like I said, institutes have started switching completely to digital workflows. So this is again very important, out of focus areas. So I'll tell you why this is an important concern, and we'll discuss some of the techniques that we have developed in our lab tomorrow. But today I'll talk about what this problem area is. So when you take an image on your smartphone, many times, I mean, smartphones are getting better, cameras are getting better, but let's say 5 years ago or 7 years ago, if you had, if you looked at some of these qualities of these images that were obtained by on your smartphones, you'd see that a lot of times because of, let's say, lighting conditions or subject movement, images would be blurred, right? And nowadays you do have deblurring algorithms on your, or deblurring let's say apps on your phone, but back in the day you didn't really have any deblurring apps. I mean, yes, you could back then. You could upload them on MATLAB and do everything and do these things that you wanted. But from a consumer perspective, blurred regions was a problem, right? And not just that. So this is something that you see on your smartphones, and we use smartphones on a day-to-day basis. So this is a problem that has also crept into the medical workflows. So we'll see how this is an issue, not just in pathology, which we'll see now, but also in radiology. Some of these applications, some of the algorithms that we have worked on, in fact, Moinak has worked on, is to de-blur a lot of these images and make it diagnosis ready. So in pathology, this is especially important because these images are huge, right? And what might happen is, because let's say maybe tissue preparation issues or digitization issues, there might be certain parts of the image or the entire image that is blurred. Okay, so I think I was asked to use this space. So you have this whole slide image here, and for some reason you have certain parts of the image that are blurred. Okay, so one of the ways. So what's the simplest way to fix it? Anyone? So you have an image. So you basically have a whole slide image. You digitized it, and so let's say the tissue preparation is okay. There's nothing wrong with that. Something goes wrong in the digitization process, and you see that some portions of the image are blurred. So what's the simplest fix? Anyone? Anyone? What's the simplest fix to?
Maybe you can pad them using nearest values, neighborhood pixels. You can pad them, mirror them, or copy them, and try to replace those pixels where it is blurred.
No, but that's completely, it's wrong, right? Because if clinicians want to diagnose these images, you can't put some other information on there, right? It has to be something even more simple than that. So there's a hand there behind you, yeah.
Is redoing it a possibility?
Redoing it, right? So one of the first things that comes to your mind is redoing it. So when our computers, for some reason you tried everything and it doesn't restart or something happens, you would reboot it completely, right? Or the old way of hitting your computer and making sure that it starts up, right? So the first thing that would come to your mind is, let's just redo it. But for some reason, digitizing it might not be possible, or let's say it will take time, right? So how do we solve that? We can think of developing and training AI models that can help us de-blur these images. So there are a lot of de-blurring algorithms that can take the out of focus regions, and based on the way they have been trained, so how are these models trained? So in order to train a deep learning model, one of the things, and there are multiple ways to do this, so one of the ways you can do this is, you can take an image, a high quality image, you blur it. So you have paired images. You have high quality image, the corresponding blurred image. You have multiple high quality images and the corresponding blurred images. So you have this paired data set, and you can learn a transformation, and I'm simplifying this by saying transformation, but you can actually train a deep learning model to understand what is the mapping between the blurred image and the de-blurred image, right? The blurred image and the high quality image. So now you can use this model to de-blur parts of the image, and now you will have a high quality image. And this image can then be used for diagnosis or for downstream AI applications. Okay. So another application similar to de-blurring or making these out of focus areas better is called super resolution. So we'll see in the next few slides how these digital pathology images are collected at multiple resolutions. Now I'll talk about that in more detail once we come to that, but just keep this in mind. So yesterday there were questions regarding site variance, right? So if there are images collected from one site, and then models trained with images collected on one side, and then you have your test data that is coming from another site, how do you ensure, what are the steps, what are the pre-processing steps that you take to ensure that your model works from images collected from another site. Right. So, yesterday we spoke about a few methods for radiologies. Now, for digital pathology, which I hinted at yesterday, one of the steps is called color normalization. So, you have different color normalization techniques that you can apply, and back then you had non-deep learning based methods, but with GIAN and with diffusion models, you have these generative methods that you can use to normalize the color intensity that helps you, analyze images that might not be in the same, let's say, color space. Right. So, for example, here's an image of bladder cancer. Your machine learning models have been trained on images that have this kind of intensity. Right. Now, what you do is, so your machine learning models, it's the other way around. Your ML models have been trained on images that have this kind of intensity, and now this is an image that you obtain, right, from another site, let's say. Now, in order to infer this, your model using this image, what you would need to do is, you would need to color normalize this image to the intensity space of the first image, right. You don't have to do that. You can always take your model and try it out and see if it works, and a lot of times people don't do that, it still works. But a recommended pre-processing step is that you color normalize it to bring everything to the same space, and once you do that, you see how the intensity of that image changes. So, this image is essentially image B that has been color normalized to A, and that gives you this C. Right. So, B is color normalized to A, and it will give you C, and you see how these two, A and C, are now, they look similar in color space. Right. So, that is something that that needs to be done before you can apply your deep learning methods. So, even while training, so a lot of times you don't have data from, let's say, a single site for training, you have data from multiple sites for training. And we have seen this, we saw this yesterday where, I told you that for the COVID-19 case, when we trained a model on Stony Brook and tried to test it on data collected from Rutgers, it didn't work, and when we went back, looked at the X-ray images, we saw that the intensity profiles were different. Now, let's say that we want to train a model together. So, we can obviously take Stony Brook data and Rutgers data, put them together and train a model, but even when we do that, we should make sure that to the best extent possible, the intensity profiles of both of them are brought to the same, let's say, space, and then we train a model, right. Now, what are the different kinds of applications that we can train AI models for? So, segmentation. So, similar to radiology, you will have different regions where you have tumors, you have interesting tissue, non-tumor tissue, you have epithelia, you have stroma, you have different kinds of glands. So, all of these primitives are important for pathologists to understand, right. So, you can actually train deep learning models to detect and segment these important regions of interest. Like I said, you can train models to detect primitives like nuclei, like glands, and so on. And what is important is quantitative histomorphometry. So, what does this stand for? So, histomorphometry is nothing but quantifying the shape, size, and arrangement of histology primitives, right. So, you can capture spatial features like orientation of nuclei, texture of nuclei, shape of nuclei, architecture of nuclei. So, all of these are important measurements that can help you in downstream analysis of different regions of interest. Also, when you look at pathology reports, you will find terminologies, right, like pathologists will tell you these nuclei are rounded or these are elongated. Now, you can always give them an automated report that would tell them, okay, this percentage of the nuclei of interest are rounded or elongated, and they can always verify that with their own analysis, and then that's how you would validate your AI model. But the reason I'm telling you this is because a lot of these features, when pathologists look at them and they give you an assessment of the shape, the morphology of the different regions of interest or the different primitives, they are subjective, right. So, they don't really give a number to that. But imagine if you had a simple you had simple mathematical filters, or you had a simple, let's say, descriptor that can compute how round the nuclei is or how elongated the nuclei is. And you can assign a number to it, that this nuclei is 75% elongated, right? So think of it as, let's say you fit an ellipse. So all of you know about what an ellipse is. So you fit an ellipse, you look at the major axis length, the minor axis length, then you look at the ratio of the major axis, minor axis. So all of these can help you quantify the appearance of a nuclei. So think of it as if we are going from a more qualitative assessment to a quantitative assessment, and that obviously has important downstream meaning, because now you can tell the pathologist that this with certain degree of certainty that this is the shape of my nuclei. I'm not just saying that, okay, there is some roundness, some elongatedness, not that. I can give them an exact number, right? So that's something that AI can help us do. So just a simple illustration of the different ways we can, or the different possibilities that we have with the current AI tools that we are developing. So cancer localization, nuclei segmentation, epithelium segmentation, tubule segmentation. So we can compute, we can basically segment out these different regions of interest that you see here. And then what can we do with the segmentations? We can extract the area, we can extract the density, the structure, texture, the spatial organization, the area, right? And then it's a lot of times you don't really need segmentation. It's okay if you can just detect the different primitives. You might want to, or the pathologist might want you to detect the where the lymphocytes are. So there are different kinds of lymphocytes, right? So these different kinds of lymphocytes mean something. So lymphocytes, like I said yesterday, they are the defense mechanism of the body. So and different kinds of lymphocytes have different contribution to the pathogenesis of the disease. So you can detect new lymphocytes, and what happens, what can you do once you detect the lymphocytes? You can actually quantify how the lymphocytes have infiltrated, like what is the infiltration level of the cancer nuclei with the lymphocytes and so on. And then mitosis, a lot of times these mitosis, so what is mitosis, by the way? There's a mic behind you.
The division of the cell.
Yeah, division of the cell. So if you look at many of these slides, you'll see that there are regions where you observe the cell division. Now there are AI techniques that have been developed that can actually help you detect what portion or where that cell division is taking place. And if you think about it, many times it can be a needle in a haystack. So these images are huge. They can be 50,000 pixels by 50,000 pixels. So the images that on your phone are probably 4,000 pixels by 4,000 pixels or even lower, right? So these are huge images, and imagine a pathologist having to traverse, navigate that entire whole slide image just to find one mitosis. Now if you can have an AI model that can, let's say, quickly go through the image and figure out where these mitosis regions are, that can essentially help reduce the time that it takes for a pathologist to study that image. Genetic subtype classification. So genetic classification is important because depending on what the subtype is, clinicians or oncologists can then decide what treatment needs to be given to the patient. So many times what happens is you have to send the tissue to a specialized lab to do sequencing and to identify what that subtype is. And this can be very expensive, this can be time consuming, and more importantly, because you don't have that, you may not even have that facility in your area. You might have to send it to somewhere else. It will take days and days to arrive. So and this can result in, the wait can result in a lot of trauma, and if you don't start treatment at the right time, this can prove fatal, right? So there's a lot of effort that's going on currently to just take the image and based on the image subtype decide what the molecular subtype of the disease is. So yesterday we saw in breast cancer, we saw the different subtypes, triple negative, HER2 positive, ER positive, right? So depending on what these subtypes are, oncologists prescribe different treatments. Now imagine if we can have an AI model that can just take the image and perform this subtyping and give you the results. So, the doctors can look at these results, assuming that these are correct, or to a certain extent if this is correct with high certainty, then you can immediately start treatment and not have to wait for the actual the results of the genomic analysis to come back, right. And more importantly, this is not tissue destructive. You have that image and you do everything digitally. So, when you do your omics analysis, you actually destroy parts of the tissue are destroyed. Now, you do not have to destroy it, you can do this analysis, and then maybe after that you can send it to await confirmation from your wet lab, right. So, that is something you can do. And then we are also using it for other higher order tasks like grading, prognosis. By that I mean, we can take these images and try to train models to prognosticate how long the patient will survive, or if there will be recurrence of the disease or not, and so on. Any questions so far? If not, we'll move to the next slide. This is a question.
Sir, you told about the like whether we are performing the staining.
Yes.
The image to image, you told, like the suppose there is a one image, we digitized it, we stained it, and then we staining the second image based on the previous staining.
You mean digitize, stain it digitally?
Digitize the first one, yeah, digitize the first one, and then second time we are digitizing it, we are taking the image, we are digitizing it and staining based on the algorithm, based on the deep learning method you told.
No, so what I said was, you digitize, so you have stain 1, stain A, you digitize that glass slide that has stain A, and then let us say that you want to have another stain. Using your AI model, you translate the stained A image to stain B image.
Yes, so my question is, sir, the staining processes for different histopathological samples are different, like it based on the receptor and ligand mechanism.
Yes.
Mostly.
Yes.
So how you are going to like how it is that much efficient that we are using one stain to
artificially
artificially doing the another.
Yeah. So the only way this can be done, or let us say a reliable way in which this can be done, is if you have enough training data with both stains, right. So, you have stain A for a tissue, and for the same tissue, or for immediately tissue right next to each other, the section next to that, you can have stain B. If you have enough training data for stain A and stain B for multiple different tissue, then you can confidently train an algorithm that can help you do the stain transfer. Okay, if you don't have enough data, it's difficult. I mean, there are unpaired generation techniques that people have been trying, but usually when you have paired data, this becomes a lot more feasible.
So in that case, we have to identify those stains,
yes,
which can be stained for both the digitized images, I guess. That will be more efficient. Like there is, suppose there are two type of stains, stain A and stain B,
yeah, and
both can be used to digitize some specific portion of the histopathological sample.
Stains are not used to digitize.
So, to stain the specific histopathological sample, and then
right,
we are digitizing it, and
yes,
then we are getting the data.
Yes, yes, yes.
Thank you so much, sir.
Yes.
So I was just curious, in terms of when you are actually digitizing the slides, for a given slide, do they do multiple, like based what you have seen, do they do multiple, like triplicates or replicates of the same slide multiple times, or just
for
once?
the same stain?
For the same, like
no,
let's say they take
just
one
once.
They do it just once.
Do it just once. But how it happens? So the internal mechanism of that is, it's not like if you, so the tissue would be probably I don't know, half of this, right? So let's say that this is the tissue, right? So if this is the tissue, you can't have that entire image taken in one shot. So what happens when the tissue is digitized is you break this up into tiles, and then small sections are sort of digitized and they're stitched together.
Okay.
And that's how they create the bigger image. But they don't do multiple, they don't do it multiple times.
Okay.
Yeah. Well, okay, so let me take that back. maybe I'm not sure about that, because if you think about it from the perspective of image acquisition, there might be some techniques where they take it multiple times and average it out, right? If let's say there is some error at the first acquisition, they might take it multiple times and average it out, but based on what I have read so far, it's just one time. Yeah.
So,
Yes.
can they build up a whole slide image on the basis of the image of different sections if they have?
Sorry?
So, suppose I have images for the different sections of a tissue. Can I use digital pathology to build up a whole slide image using the different sections of the image?
So you're saying you have a tissue section, and then what do you have?
I have images of the different sections of the tissue.
So when you have images of the different sections of the tissue, it's already digitized, right?
No, different sections of the tissues. I have slides for the different sections of the tissues.
Yes, slides, okay.
But I don't have the slide for an entire tissue.
Okay.
So can I use digitize the different sections of the tissue and use it to build a whole slide image?
Okay, yes, so you can do that. A lot of times it's actually done where, so you can read up papers on how prostate samples are digitized. So what was traditionally done is, if you don't have these glass slides where you can mount the entire prostate, they would divide it into 4 parts. They would essentially mount each of the quadrants on the whole slide image. They would digitize it, and then once you get the 4 quadrants, you would stitch them. So you would stitch them and get the entire prostate. So yes, theoretically you can do that with stitching mechanisms. Yes, and how do you stitch images? For those of you who have some image analysis background, I think you'll be knowing that. For others, so let's say that just to simplify it, you have one circle here, and you can't take the image of that entire circle at one go. So what you do is, you maybe take one portion first, and then you take the image of another portion. So you have to identify some landmarks. So this is one landmark, this is one landmark, and let's say this is one landmark, and you identify the same landmarks here, and then you stitch them. Okay, so you have something called a homography matrix. So you compute the homography matrix, and then you multiply it basically, and then with the image, and it brings that to the same coordinate space as the first one, right? So that's how you stitch images. So image stitching is a big field. So in fact, all of you have it on your phone. So what application on your phone camera has image stitching? Anyone? The panorama mode, right? So when you have the panorama mode, you essentially, what it does is, I mean, back in the day, maybe 5 years ago, the panorama mode was horrible. So as you move it, you'll see the size getting smaller or bigger. So that's called perspective warping. Now it's getting a lot better with AI. So what you do is, remember, if you notice it, if you go really fast, it will ask you to slow down. That's because it wants to take good quality images at each location. And then once it takes these images, it automatically stitches them and performs some other de-warping or unwarping operations, and it gives you a good quality panorama. So yes, you can do it. So yes, sorry.
Sir, what kind of zoom level do we have to use, like if you wanted to train a deep learning model by using
good question, very good question. So a lot of the foundation models that are out there, you'll see that these foundation models are trained at 20x. Okay. But so the reason they do that is, one of the main reasons is computational efficiency. Like if we had theoretically infinite computing power, we would do everything at multiple resolutions, because you can, when you look at the way pathologists diagnose these images, they don't really look at it at 40x or 5x or 10x. They zoom in, zoom out, they look at it at multiple resolutions. So ideally, you would train a deep learning model or a foundation model at multiple resolutions and somehow combine these features to do your analysis. But because of computing constraints, a lot of these foundation models are trained at one resolution, either 10x or 20x. Very few foundation models are out there that have been trained at 40x.
Okay.
Yeah.
Like, does it changes with the different types of cancers like that we have to give?
It can. Like, it can. So there are papers that have indicated that certain resolutions are better for diagnosing certain diseases and other resolutions are better for diagnosing other diseases. If you have some prior information like that, it's always good to start training your model with that level of resolution which is good for the particular disease.
Yeah. Thank you, sir.
Yeah. Okay. So, as a summary, what are the reasons why we want to use AI in pathology? First, time consuming and cumbersome tasks like manual annotations and cell counting, all of these can be reduced. But the primary idea is to have reproducible measures. So, if you tell like with certain certainty or with conviction that there are round nuclei or elongated nuclei in that particular tissue, then because you provide numbers, when you provide it to pathologist A, B, C, D, they see the exact same thing. Otherwise, what you have to tell them is very subjectively, very qualitatively, you tell them that, okay, you have approximately, half of the tissue is rounded or nuclei is rounded or something like that, which doesn't really help from a reproducible purpose. And there are these subtle image patterns that may not be discernible by human eyes, which where AI can really help. And the ultimate objective is precision medicine by discovering new diagnostic and prognostic biomarkers. So now let's come to what are some of the classical handcrafted features in digital pathology. So yesterday we saw in the first part of the lecture certain radiomic features. You remember collage features which were used to study the morphology or the texture of the brain tumors. So similarly, a lot of research has been done to study how handcrafted features in digital pathology can help in diagnosis, prognosis, and treatment response assessment. So here I'd like to also tell you all of you that nowadays we are beginning to move away from handcrafted features. With deep learning, we've seen that it's much more powerful. The results are more accurate, better when it comes to things like tasks like detection of disease, diagnosis of disease, prognosis of disease. Typically, handcrafted features, even though those can be understood by experts, by pathologists, the performance of many of these features are not that good. But having said that, what we should understand is that let's not completely discount the handcrafted features, because these handcrafted features have an intrinsic meaning, right? And I'll tell you about what these handcrafted features are in the context of pathology. But what we need to keep in mind is that there has to be a way, or we should come up with ways to retain the improved or the increased accuracy of deep learning methods, but at the same time not lose the interpretability that some of these handcrafted features can bring to the table. And we'll cover that in 2 lectures tomorrow. Okay, so by the way, the question that we want to address here is, can quantitative histomorphometry of the tumor morphology, can they predict disease aggressiveness and patient outcome? So what you see here are 2 samples of just part of the whole slide image, and here are the different kinds of features that we can extract. So we can construct graphs, and there are different ways of constructing graphs. You can have Delaunay triangulations, Voronoi methods, minimum spanning trees. So you can have graphs, you can have grayscale features. So grayscale features, by that I mean intensity based features. You can have texture features. And so there was this question about what magnification, right? So pathology images are collected at multiple different resolutions. So you go from 5x all the way to maybe 40x, 100x, 120x. So at different scales, you observe different structures. So let's go back to the previous slide. Here you can see in detail where the different cells are. Now if you come out one level, you can begin to understand that cell that region of interest from the previous slide was probably somewhere here, right? So now you do see cells, but you also observe the context. You see, thanks. So you also observe the context. So you see what are the different kinds of cells around it, right? So it's much more information you see as compared to just a small patch. We don't stop there. We can further zoom out. We can create graphs. We can look at the intensities, and then now you are beginning to see what the whole slide image looks like, right? So that small image that we saw in the beginning of the slide is probably one tiny speck here, right? So you see how we are getting more and more context, and this helps us it's very similar to, let's say, what you have in your phones, the camera, the photos that you take using your camera on your phones. Even though the way of storage, the storage mechanism is different, you have a pyramidal format here, you do not have a pyramidal format there. You see, if you zoom in and zoom further into your images on your smartphones, you'll see it gets blurry, right? But if you zoom in or zoom out of the image that has been collected using a digital pathology scanner, it is not blurry. It will still maintain that level of resolution. It will still look, the resolution will still be very high because of the way these images are collected and stored. And then you can create, so the first slide we saw the graphs, those are more local graphs. These are more global graphs. So think of graphs as, let's say, connecting, the centroids of the different nuclei using some logic, right? So let's say that if the nuclei are round, then you connect them, all the round nuclei connected to each other, all the elongated nuclei connected to each other, you form a graph. So you can do that not just at a patch level, but you can slowly start to zoom out and construct that graph at a whole slide level. And this is what you see here. And so a lot of these measurements, these graph based measures can be used for downstream analysis. Further down, you see how we now see the entire whole slide image. Okay, so now pathomics, so like we saw radiomics yesterday, so pathomics is nothing but the science of extracting features from digitized pathology images. So these features focus specifically on the section tissues, and the scanners, like we saw, they create the high resolution images that can then be digitally processed. So you can identify different kinds of cancer, you can annotate, you can look at multiple different resolutions. And what are the different features that you can extract? Shape-based features, right? So shape-based features, so this is the just a small tile from the whole slide image. You can extract features like shape-based features where you are looking at how round the nuclei is, how elongated the nuclei is. You can look at the architecture. By architecture, I mean how they're arranged with respect to each other. If you have one tissue type infiltrating into another tissue type, that means something, right? There are different levels of infiltration. All of this you can compute and you can assign a number. So a lot of these can be made quantitative. And then texture. So similar to what we saw yesterday with the Haralick features or the collage features, you can apply the exact same mechanism to these nuclei. So once you segment out the nuclei, apply your Haralick features, apply your collage features, and get the texture values. And this is how the the texture of the nuclei would look like, right? And why is this important? So let's. I hope you can see this on the screen. So look at this particular nuclei here. You see how it's dark, and then look at another nuclei here. So you see how it's a little bit brightish. So when you compute the texture feature, you'll see that the texture response of the nuclei here will be very different from the texture response of the nuclei here. And then you can actually use that texture response to maybe construct a feature vector for downstream classification analysis, right? Okay, so what are these different textures and what do they quantify? So a lot of times, because of different kinds of mutations, the texture of the nuclei might be different, or certain kind of texture might be associated with certain kinds of mutations. So that's why it's important to study these textures. For example, in this particular case, you can see that the texture is different, and these kinds of texture led to good prognosis of the patient, and this texture led to bad prognosis or poor prognosis of the patient. So what is the first step before most of this analysis? You have to segment out, you have to detect and segment where the nuclei are, and after you have segmented it, you can apply the different features. One of the very common features, like we discussed yesterday, is the Haralick features, and this is the entropy of the so Haralick has about 13 or 14 different features. One of the features is the entropy features, which quantifies the disorder of the pixel intensities, and if you compute that, you see how these filters, how these responses are quite different. And yeah, so let's go to the other feature, shape feature. So one of the simple shape-based features that you can compute is nuclear size. And how would you compute the nuclear size? So what you do is first you have a deep learning method or some method to extract where the nuclei are, and then simply you compute the number of pixels. So if you have a large nuclei, the number of pixels would be high, right? And if you have a small nuclei, the number of pixels would be low. And you see this is a much larger nuclei here probably, I mean it's part of it is cut off, so you have a high number of pixels. So simply counting the number of pixels within the segmented region within the nuclear contours can give you the size of the nuclei. So solidity of the nuclei. So this is an interesting feature where you want to quantify how solid the nuclei is. So let's say that you have a nuclei that looks like this, and you have another nuclei that looks like this. The first thing that you do is you construct a convex hull. So this convex hull would possibly look like something like this, and you construct a convex hull here which would possibly look like this, right. So you see that, so what is solidity? So solidity is nothing but proportion of the pixels in the convex hull that are also in the region, right. So here's the nuclei, I am shading it here. So clearly the proportion of this nuclei that is inside the convex hull is much lower as compared to this nuclei, right. So the second nuclei that you see here is completely, it's very strongly associated with the convex hull, but the first nuclei you see a lot of gap between the nuclei and the convex hull. So which means that the second nuclei is more solid as compared to the first nuclei, right, the first nucleus. So these are simple ways of quantifying the shape of the different nuclei on the whole slide image. Now another feature, like I was telling you, is axis ratio. So you can fit an ellipse to the different nuclei that you have segmented, right. So once you fit an ellipse, so let's say that this is a nuclei, you fit an ellipse to this, you have a minor axis, you have a major axis, and one of the very simple features can be the length of the major axis, the length of the minor axis, and the ratio of the major axis to the minor axis. So these can be simple yet powerful descriptors for describing what the different nuclei in the images are. Okay, now going from shape based features to architecture based features, first what you do is, first of all, why are architecture based features important? So these the histological architecture it's basically used, or you can use the architecture based features to quantify the structure of healthy tissue and the structure of diseased tissue. Now if you have distinct structures, they usually indicate healthy tissue, and if you have loss in structure, it indicates uncontrolled growth or cancer. Now if we compute graph based features, you see that for the healthy tissue you have more well defined structures on the left as compared to the cancer tissue, where you have a different kind of loss in structure, right. So the differences in structure can then be quantified and can be used to decide whether a tissue is cancerous or not. So there are different kinds of architectural graphs that can be extracted. So Delaunay triangulation, Voronoi diagram, minimum spanning tree. So doesn't matter what algorithm you use. So the first step of constructing these graphs is that you have to identify what the nodes are. Now for the nodes of the graph, there are different ways you can do that. First you segment out the nuclei. So after you have segmented out the different nuclei, you can take the centroid of the nuclei and consider that as a node. Or, so that's a nuclei graph. So let's say that you are interested in a gland graph. So what you would do is you would segment out the glands in the image, and the centroid of the gland can be the node. So depending on what kind of graphs you want to construct, you can segment out the primitives, identify the nodes, and then based on different rules that you have, you can construct the edges of the graphs. Okay, so what else can we do with this? So there is this notion of compartment aware analysis. So it's not necessary that you have to analyze the entire tissue in one go. So because these are huge images, there are different compartments in these images. You have epithelia, you have stroma, you have different kinds of cells. You have regions where you don't really have any tissue altogether, right? So you don't have any tissue in some of these regions for whatever reason. I mean, there can be tissue loss during the preparation, or for some reason there might be some hollow regions there. So what we can do is, you can have algorithms to separate out the different compartments and then do a compartment specific analysis, that this is what my stromal features look like, this is what my epithelial features look like, this is where you have the tumor infiltrating lymphocyte deposition, this is what my TIL features look like, this is what my healthy cell features look like, right? So you can divide your image into different compartments and do a compartment specific analysis. So this is the stroma, this is the epithelia, and you can see how the stromal cells and the epithelia cells have been color coded differently. So you can do your analysis like this. So different kinds of deep learning methods have been developed for analyzing these images. You can have methods like segmentation methods, like I said, where the method. So one of the commonly used segmentation methods is called HoverNet, horizontal vertical network. So this code is available and pretty widely used. So this was published by Dr. Rajput's group from Warwick University. Since then, so this was the pre-transformer era. So in the transformer era, you have methods like CellVIT. So these are methods that are much more powerful, and they can not just segment out nuclei, they can also assign the nuclei into different classes. Right? So you can have different classes of nuclei. Mitosis detection algorithms, like I said, cell counting. So once you detect the cells, you can count the cells. But the challenge is that because these images are so huge, you would need large training data, and a lot of times, yes, you can develop these methods, but there is not much interpretability at the end of the day. But we'll see how we can address the issue of interpretability in tomorrow's lecture. So tumor identification, classification, anatomical region classification. So a lot of times the analysis focused on small patches because of computational challenges, but there are new methods like multiple instance learning. So using multiple instance learning methods, we can take patches, smaller patches, and combine them to come up with a reasoning for the entire slide. Okay, so maybe 10-15 years ago, most of the papers that came out, you would see that the analysis was restricted to very small portions of the image. But now, we know that if we do that, we are losing so much information. There's so much available information in other regions, other parts of the image that we are losing. So there are methods like multiple instance learning, like I said, that can be used to come up with or arrive at decisions at a whole slide level rather than a patch level. So here are a couple different papers. So this paper that you see on the right, so there's this concept of superpixel or supervoxel. So what does that mean? So you have, what you can do is you can divide you have pixels. So pixels are essentially each individual unit of the image, but you can look at pixels right next to each other. They share the same, let's say, similar intensity information, and the pixels that share the same intensity information, you can club them into one group. You call it a superpixel. So you take your image and you construct these superpixels. So you see this honeycomb-like structures here. So these are all superpixels that you see here. So all the pixels inside the superpixel share some similar information, whether it is intensity or some, let's say, orientation, entropy. So you construct these superpixels, and instead of doing your analysis on the pixels, you can do your analysis on the superpixels also. So that's something that can be done. So in this particular paper, the whole slide was partitioned into superpixels, and after that they did a graph clustering to group the anatomical regions, the pixels into the superpixels into different anatomical regions. And based on that, you can then run a classification to see if your superpixels belong to the cancer regions or the benign regions. Okay, so this is work from some colleagues from our group, Dr. Joel Salz, where the goal was to identify tumor infiltrating lymphocytes and then generate this spatial TIL map. So it has been shown in multiple papers that having good concentration of TILs leads to even good prognosis eventually. So that is why it becomes very important to identify where the TIL regions are. And once you identify where these TILs are or the lymphocytes are, you can essentially assign numbers to these patches. Okay, this patch has 98% TILs, this patch has 50% TILs. So you can construct a TIL based map, a TIL map, but so which looks like, so this is a cancer map and this is a TIL map. So you see that here you have high concentration of TILs, here you have high concentration of TILs. And once you do that, you can provide this to a pathologist, and the pathologist can look at this and say, okay, this image looks like it has heavy TILs. This means that this person might have a good chance of prognosis if given a certain treatment, right? So you can perform this analysis, and in addition to showing the pathologist this image, you can also give them this image. Right. So yes, they can do that, they can look at TILs, but they can obviously not come up with a map like this immediately. But if you give them a map like this, they can look at the red regions, they can verify first of all whether it is correct or not. Assuming that the algorithm is well validated, they can look at your TIL map and they can tell you what sort of treatment, or they can tell the oncologist that this is the TIL map that we have for this particular patient, which means that this person would likely benefit from therapies like immunotherapy. So let's go ahead with that. I mean, the pathologist won't make this decision, the oncologist will, but the pathologist at least has the supporting information that we provide to them in addition to the digitized image. So this is work from our group, and this actually this was published 3 years ago at SPIE. So here we were looking at classifying images into 3 different groups. These were brain cancer images. So astrocytoma, oligodendroglioma, and glioblastoma. So we had the whole slide image. What we did was so this was before multiple instance learning. So we identified patches from it. We took features from each of these patches. So this was at a low magnification. So at a low magnification, we identified which of these patches would be important, and then looked at only those patches at a high magnification to do the analysis. So that's what we did in this paper. But nowadays, we have moved on, and we look at whatever magnification we want to, but look at the entire image. We don't pre-select important patches. That is what multiple instance learning actually gives us. It gives us an attention weight for each patch. It tells us which patch was eventually important for the downstream classification. You don't have to pre-select patches. So why else or where else can we use these digitized pathology images for multimodal integration with other cancer? So there's a lot of research that has happened in RAD path registration. So here's an example of a prostate scan. This is a radiology scan of the prostate, and this is the corresponding tissue section of the entire prostate. So there was this question that was asked, what if you have different sections, can you put them together? So this is what I meant when I was explaining to you the different quadrants. So previously, if you did not, when we did not have the capability to digitize the entire prostate, they would actually take these 4 sections, digitize them separately, and then stitch them to get this entire prostate. So now that you have the prostate sample, digitized prostate sample, you can also, let's say that you have the image of the prostate that was taken before the prostate was removed from the body. Now you can have a registration method that you can use to register the MRI onto the pathology or the other way around. Now what this can tell you is which part of, so let's say that you had this part of the image as cancer, that would correspond to this part. Now you can study what is the radiology characteristics, what are the radiology features associated with this part, and what are the corresponding pathology features in this part. So once you understand, or once you have the features coming from different modalities, you can do some sort of a correlation analysis, associative study to see how features at one length scale are related to features at another length scale. So there was this question yesterday, like, okay, how do you even understand what's happening, what's the underlying biology? So you get something in the radiology length scale, what's happening in the pathology length scale. If you have mechanisms like this, where you can co-register images or radiology images onto pathology images, then you can identify the exact same area where where tissue are corresponding to each other and study them in great detail. So, not just images. So, similar to radiology, pathology images also have reports, right. So, we can extract reports and then we can use the reports to do multiple different things. So, this slide that you see here will come back to this tomorrow, but what I want to say here is what we did here in this particular project was we took the images, we extracted different kinds of features like TIL based features, tumor based features, but we also took the corresponding pathology report, we provided it to a large language model, and in this case it was GPT. We provided so the model gave us a summary of what the report had. So, okay, why did we want a summary? So, I do not think we will go into the details of this, but we wanted a more concise representation of the report. Like the report will be long, but we did not really want a long report, we wanted something short and a concise report. So, once we had this concise report, we provided it to a language embedder, we got the embeddings, and then we had the image based patches that we got the embeddings from that, and then we used this conditioning mechanism to a model, to a generative model to help us generate synthetic samples of that image. So, we'll come back to this and discuss this in more detail tomorrow. So, but the bottom line is what I want you to take away from this is there's multiple modalities of information within this. So, we have digitized pathology slides, you can have different kinds of stains like we discussed, it is not just H&E stain, you can have DAPI stain, you have Key 67 stain, different kinds of stain, and then also you have the pathology reports associated with those slides. So, it is not just imaging and pathology, you can also combine genomics. Obviously, you have genomics information corresponding to those, you can have proteomic information, you can have immunohistochemical features, and you can combine all of this information theoretically to get better classification, prognosis prediction, and treatment response assessment. So, ultimately what we want to do is we want to combine histomorphometry with molecular data for more holistic disease characterization, and that would lead us a step closer to precision medicine, which is essentially tailoring a treatment plan to each of the individual patients that we have using data from multiple different sources. So, what are the different challenges? Obviously, interpretability. So, like I said, a lot of times we are beginning to move into an era that is completely deep learning dominated, which is important because we have a lot of computing power, and we have seen that if we can apply these deep learning techniques, the accuracy of these different classifications increase, but it comes with a risk of decrease in interpretability. A lot of times interpretability does not really matter. For example, if the goal is to just segment the nuclei, right, just to segment the glands. If your model does well, your model does well. We do not really care about how that model works, or the doctor will not really care about how the model works, as long as you say that, as long as your model gives you the right regions of interest. But if it comes to treatment response prediction, and your model tells you that this patient will survive for n number of years and this other patient will survive for p number of years, if n is much lesser than p, right. So, then the doctor will want to know why did the model sort of give you this prediction that this patient will survive for fewer number of years as compared to the other patient. So, that is where interpretability becomes crucial, right. Color variations, we covered that, so that can lead to issues in training and testing, and we need to make sure that we do our due diligence to ensure that this color variation is accounted for in your model training and inference. And so, these slides that you saw here, they are all 2D in nature. So, yesterday the 3D images, the MRI images that we saw, they are actually 3D in nature, right. So, you have the brain, you take the MRI of the different sections. So, it is a 3D image intrinsically. So, even though the tissue that you extract from these histology images, they are 3D tissue because it is not just one slice, right, you will extract a bulk of it. But when you section them, these essentially become 2D sections. Now, there is a lot of research now going on in 3D pathology. So, you can actually take the 2D sections and stitch them together to form a 3D histology, digitized histology image. Now, what it can help us do, So if you reconstruct or if you stitch these images together or layer these images together appropriately to get your 3D histology sample, you can actually look at structures. So there will be blood vessels or there will be some other structures which you can start to observe in 3D, right? So we are moving from, there's a lot of research that's going on where we are moving from 2D to 3D analysis to get the depth information. Multispectral images, it's not just a single image, but there can be multispectral images. And last but not the least, we need high performance computing because of the high data. So this is truly big data. So if you look at one whole slide image, it can be anywhere between 1 gigabyte to maybe 3 to 4 gigabytes. So these are huge images, and a lot of times it's very impractical or almost impractical to load the entire image into your RAM and do the processing, right? Because you'll have to, it's not just one image, you'll have to do them in a batch, you'll have multiple different images. So there are methods that people are developing to see how these images can be loaded onto your computer memory in order to make the processing more efficient. Okay, so I think that's it for the first lecture of today. Any questions?
What are multispectral images?
So by multispectral, what I mean is, it's similar to if you have multiple different channels, right? So let's say that you have one stain and another stain, you can look at different channels of the same stain, and this basically become multispectral.
Sir, we have seen that we are using now deep learning techniques to extract features and automatically classify images.
Yes.
And sometimes you mentioned hand-captured features are not useful. So I have in mind, like in some applications where two different classes have similar textures and everything,
yes,
and the model fails to classify, there is no clear-cut boundary. So how can we combine efficiently this along with deep learning and hand-captured features? Is there any way?
Yeah, we'll see tomorrow in tomorrow's lecture. Yeah. But I can give you a teaser. So let's say that you have a deep learning model and you have your handcrafted feature model. So what we can do is we can train a deep learning method to learn from both handcrafted features and the actual images. So if we come up with a co-training pipeline, we can basically use we can leverage the high accuracy of the deep learning methods, but at the same time retain some interpretability coming from the handcrafted images. But we'll see that architecture tomorrow. Yeah. And it's not just handcrafted features, right? I mean, yes, handcrafted features are, end of the day, handcrafted features are something that we craft as as scientists for the pathologists. But what is probably more important for them is if we can use some language based features, right? So what if you can say that, okay, there is in exactly this, in this terms, you can have a sentence saying that, okay, X nuclei or tumor infiltrating lymph, there's a heavy infiltration of cancer cells into the TIL region or the other way around. So these kinds of descriptions are better understood by the pathologist. So if we can train a model, a deep learning model that can take into account these descriptions and use these images, and when you provide the output, you can say, okay, this is a cancer because of this concept. That will be more important. So that's also something we'll see tomorrow.
Any more questions?
Okay.
I think that's all. We'll wrap up for now. Just have your tea outside, and then we'll let you know how the next program progresses. I will inform you. Well, there was one more lecture scheduled, but we'll discuss with the expert and the team, and maybe we might have it after lunch or something. We'll update you about it. Okay. Thank you very much.
Thanks. So, Mirza, do they want to put
record. हेलो रेडी ऑप्शन प्रोजेक्टर ऑन कर
ठीक है हाँ कर
जस्ट मिनट
Okay, good afternoon everyone. Welcome to the second day of our GIAN course. This is the first hands-on experimental session. So far we have had 4 lectures, and in today's hands-on experimental session you will get to experience all that you have learned through the lectures. So today's hands-on experimental session actually has been prepared in collaboration with Professor Prasanna and his team. So in this hands-on session we will actually start from medical imaging 101, so introduction to medical imaging. Then we'll move on to how to handle medical imaging data. So for this there is a software which you will all get to use. You can check or at least you will see how this software works in action, 3D slicer. And then we'll look at basics of pyridiomics. This is a package that you will be using through the Google Colab. There is an environment, there is a using your web browser you can use Google Colab, and Google Colab allows you to access GPU from Google servers and run your own code without having to install any software on your own laptop. And then we look at basics of pathology informatics, and finally the big chunk or the major part, which is the foundational models. So nowadays a lot of people are working towards development of foundational models for different applications. So you look at foundational model development in relation to medical imaging. So with this I hand over to Professor Prasanna for further details.
Thank you. So I'll just quickly to go over some of the topics that Monak will cover in more detail. So in introduction to medical imaging, what we'll have, so we saw how we can apply, we can extract radiomic features from images like MRI scans, or if you remember the lung vasculature extraction that was done on the CT images. We covered a couple slides on ophthalmic imaging, if you remember, so the ultra wide field fluorescent angiography scans, and also for the dental imaging we had CBCT scans. So these are all different kinds of imaging modalities that you will encounter when it comes to working with radiology data. So obviously the most common modality is the X-ray modality or the radiographs. This is an example of a small cell lung cancer CT scan. So you'll, over the course of this tutorial, which will last all of today and possibly tomorrow, depending on how much we can cover today. I mean, we have other topics that we have lined up for tomorrow as well. But you'll learn to load these images. First, let's say that you have a data set. How do you load these images? How do you view these images? So there was a slice. There's a software called 3D Slicer. There are other software like ITK Snap. I don't think we'll need you to download this software right now. So we already have that on our machine, so you can see how to work with those. So software like 3D Slicer, and Monak will go through it in more detail. So you'll see you'll see how you can not just upload images, view images, but these softwares also provide other basic functionalities, such as if you have a tumor, you can upload the mask, or you can overlay the mask on the image and see where the tumor is, where the boundaries are. You can also do some very basic measurements. In fact, you'll also learn about pyradiomics, what we had in the topic slide. So what pyradiomics is essentially a library that you can use to extract radiomic features. So it has a bunch of radiomic features. You can just it's wrapped. It's sort of a wrapper that you can use to call and then call the functions to extract these different kinds of features. We won't really cover a lot of classification examples today. So those are the ones that we'll keep for tomorrow. But you'll see how to play with how to invoke the pre-trained weights and all of that for many different things along with the foundation models. So I haven't really covered much of what foundation models are. So I mean, foundation models is essentially it's a new terminology. It's something that's thrown around nowadays. All that it means is models that have been trained on lots of data, right? So instead of having models that is very specific to or very localized to your institution, maybe hundreds or thousands of data points, you have millions of data points that these models have been trained on. So those are the foundation models. And it's not necessary that if you download a foundation model off the shelf, it will work for your data, and most likely it will not. It'll work in the sense that it'll give you some results. It might not be the best results. Usually foundation models are fine-tuned further for your own purposes. So with that, yeah, so these are other kinds of images like PET images. Like I said, we'll look at the tumor segmentations, other objects of interest. We'll also look at pathology images. So if you remember earlier today, we covered digital pathology and how you can open these digital pathology scans. These are huge images, so bear with us. It'll take some time to actually load these images because we don't really have the specialized software for that. You'll have to use, we'll still use Python, right? Yeah, for doing this. And yeah, so with that, I'll hand it over to Moinak. So by the way, for those of you who don't know Moinak, so he's soon to be Dr. Bhattacharya. So he'll be defending. He's in India to get married, but I had him come over because he's sort of used to working with this data, and he's aware of some of the latest models that are out there. So I thought it would be a good idea for you to just interact with him. And also beyond this tutorial, feel free to reach out to him. So he's about to start a postdoc right now, and he'll be soon in the academic job market. So he'll be able to also talk to you about what are the prospects out there when it comes to getting jobs in medical image analysis or staying in academia. He also has some industry experience as well. So yeah, Manak, over to you. So by the way, everyone has their laptops ready, right? Good to go? Cool. So at any point, if you have any questions, feel free to stop Moinak, and then we'll try to address your concerns.
Thank you very much, Professor Baig and Professor Prasanna for the introduction. So let's start with some introduction to medical imaging. I hope you guys have the Google Colab drive or the drive that has all the Google Colab files and the images, right? Okay. So in the file there is one IPython notebook named NITR Session 1 Basics. So if you just download it, I can just give a brief tutorial on how to download this and upload it in Google Colab. So it's simple, just download it. It will be downloaded in your downloads folder or whatever folder you have set to your system. For example, I'm downloading it in my download folder. You can go to any browser, just type Google Colab and welcome to Colab will pop up. Just click this and you can click this new notebook option.
Here. Can we switch to white mode
Let me try, 1 second. How about now?
This is better, but maybe just increase the font size. Do control command plus. Yeah, better.
Okay. Is it visible now? Okay. So I hope we have a Colab window open. You can go to File, click Upload Notebook, and then click Browse, and then wherever you have downloaded this basics IPython notebook, click Upload, and it will take some time depending on the internet, and you will have the Google notebook uploaded. Okay, okay, sure. Okay, sure. Okay, sure. So are we stuck anywhere while uploading the Google Colab?
All of you have logged into Google Colab?
And are you able to upload the basics file? Okay.
If you are not able to do it, please raise your hands. We'll try to.
Please raise your hand if you are not able to log into Google Colab.
Yeah. Meanwhile, people who were able to log in, because this step takes some time, just click this folder button, this folder, and if you click this, this will restart, or this will start your Google Colab session. So something like this will pop up, and click this upload. Meanwhile, also download this chest X-ray image from.
First of all, download that chest X-ray image from the drive link.
Or you can do one thing, right click on that file and open with, there is a term, and then directly you can open with Google Colab. You don't
Yeah.
have to download it.
Yeah, that also you can do. Sometime what happens is if you are logged in through your institutional email, it might not support Google Colab, but if your institution supports it, then just right click Google Colab and open it.
You just open that drive link and then right click on that file. You will see that like open with Google Colab. Okay, so it will directly open. You don't have to download and open it.
Yeah.
So that will be easy for you, I guess.
Yeah. You can click this open with and then open in a new tab.
So Mahesh and Somajit are going row by row. So if you have any issues, you ask, just raise your hand, please. So we'll go a little bit slow in the beginning,
Yeah.
so that you all catch up.
Sure.
And then. Sir, any issues? It's already open. Just use your own email. Okay? Our email is not. Use your Gmail. Are you all in your? Have you all signed in with your Gmail? Yeah, then click the drive link. Then you can open with. All of you are able to get the drive link. Can you see images inside the drive link? Password problem or you are not password problem? Okay, fine. Okay, so after the, so do they have to upload or what?
Yes, so download the chest X-ray image and this TCGA A62686 file. Download this, and then once you are here, wherever you have downloaded it, just upload them. So just click that file and click upload. It will give you a warning, just say OK. And same goes with the TCGA file, just upload it. So the TCGA, this file is a histopathology image. This is a big file, so it might take some time. Yeah, if you have if you have time, download all the files in this folder.
First of all, download all the files. Yeah, we have ample amount of time, so we have around 3 hours, so don't worry. And if not, but we don't want you to miss out, so that you can actually see how things are working. So
Yeah.
please download the file from the drive link. Downloaded. All of you have downloaded? Front row, maybe they are waiting.
Yeah.
Downloaded? Yeah. So we'll wait for you, don't worry.
It might take some time because there are histopathology images and brain MRIs, which might take some time to download.
So after you have downloaded, you can open Google Colab. So if you are logged in into your Gmail, you can simply go to your browser, type Google Colab, it will give you a link. Or you can do Google colab.research.google, there is some link.
It's 475 MB. Please use the
Wi-Fi.
Don't use a mobile. Just kill the time.
Okay. So if you have downloaded, go to your Google Colab and then upload, right? Which any specific file they need to upload?
No, so as of now, you can upload the chest X-ray and the TCGA A6 ... this file. So the way to upload the file is click on this up arrow button, hover to the folder that has the files in it, click the
Yes.
files, click upload.
So on the left hand side, you will have a space, yeah, like that.
So yeah,
Just check bench by bench.
It's the Jupyter notebook file. Don't think that anything else. Okay. So and download the 2 data files.
If you have downloaded the files, please upload it in your Google Colab window. If you are not able to do it, please just raise your hand. We'll come and help you out.
yeah. These are the critical steps. So if you are not done through this step, we are not moving forward.
Chest X-ray is very small. Just download it and upload it. It'll take barely 1 minute.
Let's check all they have done. No?
You uploaded, right? You uploaded. There is no problem.
Test image is that will not take much time, but the SVS file will take some time. So if everyone has uploaded the chest X-ray image, we can move forward.
No, just wait, please.
Oh, okay.
Yeah, few people are here.
Yeah, sure. Take your time.
So the files are a bit large, around 400 MB. You can use your Wi-Fi. Don't use your mobile net, and please download. Once you have downloaded, you can upload in the collab window. We'll wait for some time. So if you are getting that slide, like you can see there are 3 lines. Sometimes it's not visible. You have to press that.
Yes.
Yeah. Sometimes it's hidden. If you press and then you can press the upload button. Yeah. Please upload the chest X-ray image. Please upload the chest X-ray. Yeah, doubt.
Hello. That section is called data uploading section. At there you have to upload that image, not in the like a full website. There is a small option. There you can upload that image. There you can upload your data. That image is treated as your data, own data. Okay.
Sorry, folder. Yeah, that folder.
It will come. It's connecting. No? It will come. And after that you can see that option. Small option, then you can actually it's connected.
If the TCGFI downloaded that.
Yeah. So you can see on the left hand side, for some people it's not visible, that files main option is not visible. So on the left hand side there is a folder icon, 1, 2, 3, 4, 5. folder icon. Just click on that folder icon.
Yeah, it might be hidden like this. You have to just click it.
Yes.
And then you
Press
will
upload.
get this upload folder.
So upload will also take some time. So we'll wait for some time. No worries.
The TCG upload can wait, but the chest X-ray upload should be there. The TCG can wait. We have that in the later section of this notebook.
So first upload the chest X-ray image. It will be faster. TCG you can do it later.
Yeah, no, you start uploading. We'll use that later. It will
Yes.
take 5-10 minutes to upload.
Problem. How many of you are still uploading? Just raise your hand, please.
The chest X-ray.
Chest X-ray. How many of you are still uploading? Okay.
Okay.
Few people are still there. Just upload the chest X-ray. Chest X-ray and the TCG also you can upload, but it need not be 100% uploaded. Just the chest X-ray is the first experiment, you can say, or the first code that we are going to look at. I think. You can follow on your friend's laptop. It's okay. No worries. You can follow on your friend's laptop. So all of you uploaded the chest X-ray. Next step, please.
Okay.
Please listen.
Let me start with some basic discussions about what are machine learning, medical imaging, and why do we need this. So the first modality that I'm going to talk about is chest X-ray or X-ray in general. We know, since our childhood, why an X-ray is needed. It's mainly to check fractured bones or sometimes pneumonia, like the first step if you have dense accumulation of fluid in your chest. So generally that's why we need a chest X-ray. A CT scan, it's so chest X-ray, it's a 2D image. So I don't know, maybe if all of you might have gotten chest X-ray at some point in your life. So you will just stand either facing or putting your back towards the receiver. You will have an emitter in the front. The X-rays will come across your body and the receiver will receive it. So it's a 2D modality. Whereas CT scan is a 3D chest X-ray. I mean, in basic terms, they will make you lie down and the 3D the receiver and the emitter will pass through your body. And MRI is also similar. It's a 3D imaging modality. But can anyone say, I'm assuming most of you are BMI, biotechnology or whatever in the related fields. What's the basic difference between a CT and an MRI? I mean, very basic, I don't think you need a very high level understanding of medical physics, but what's the basic difference of MRI or and CT?
Please use the mic.
Based on X-rays and MRI is
in CT mostly means we are using X-rays, so which gives image based on the different absorption, different tissues absorb different wavelengths, and in MRI it's based on the principle of magnetic field, and means there is a strong magnetic field in between V goes, and then a radio frequency pulse will be applied. So its hydrogen atoms will excite, and when we apply radio frequency pulse, it will come down to the normal state, and it gives us what signals.
The K space of that magnetic field will be stored, and then we use that K space to reconstruct the MRI.
Yes,
exactly.
yes, right.
Thank you. So, and another difference between a CT and an MRI is CT has radiation, whereas MRI does not. So it's safe to have brain MRI scans using brain scans using MRI. Another very interesting modality is ultrasound. It's generally used to do some real-time imaging of the heart or during pregnancies. The only disadvantage of ultrasound is it's very much the technician dependent. I mean, like a very inexperienced person might get a very low quality ultrasound. Maybe for a radiologist it will be not of that great diagnostic quality, but a very good technician can take a very great ultrasound, and also again no radiation. There is another interesting imaging modality. It's called PET. Any idea what's the full form of PET? Positron emission tomography. It's mostly used to capture the metabolic and functional part of the imaging, and PET is generally acquired in conjunction with the CT or MRI. So it's called PET CT or PET MR. So in this let me go back to the slide, and here I can this is a chest X-ray of the thoracic region. This is a brain MRI where we can clearly see a tumor. Any idea which modality is this? So MRs generally have 3-4 modalities, the protocols, the sequences. Sorry, the T1 post contrast, the T1, the T2 weighted, and flare. So any idea what modality what sequence is this? T1 what? So this has contrast. You see there is contrast here. So it's T1 post contrast. This is a lung image of a patient with small cell lung cancer, and this is what I was talking about.
Can you go into slideshow mode, please?
Oh, sorry. Yeah. And this is a PET imaging. The left side is the PET, and the middle one is the CT, and this is the PET CT registered, where you can see the glucose decomposition happening. So here and the brain has higher level of glucose decomposition. Generally for PET we measure the glucose decomposition using SUV, that's a metric. Okay, so and PET is generally very useful to detect tumors, or to detect very small tumors which are not very readily visible by naked eyes. Okay, so now we will work with some libraries which we generally use to upload medical images. So here, here I already have written the code to install some Python wrappers, PyDICOM, Nibable, OpenCV, Matplotlib, and scikit images. So PyDICOM is so when we acquire a medical image, we acquire it in DICOM format. So Python created a library which eases our way to upload, download, save an image, and work with DICOM images. We call that PyDICOM. And then there is another way of storing medical imaging modalities, that's called the NIFTY, the NII.gz. And Nibable is another Python library which is very handy to upload and download and work with NIFTY files. So to run this Colab cell, you can either click this run button, or you can just press shift return or shift enter, whatever works, and this will download or install all your dependencies. Already done? That should be fairly easy.
All of you have done that step? Yeah? Done? Working? Done? Fine?
Yeah. So just a quick yeah.
Done, right?
So just a quick suggestion, we are installing all these things in Colab very easily. Don't try to do this in your local system. It will just mess up with your environment. The good practice is if you are working on, if you want to run something on your local computer, it can be a Windows or a Mac, or doesn't matter, install Anaconda. Create an environment, a Python environment. It's recommended to create an environment with the updated Python version, and then in that do all this experimentation that you want to do. Please don't do it in your local system, or you can use Google Colab. It's very easy. Okay, so we uploaded. I already discussed what SpyDICOM, Navable, etc. Now the first step is loading and displaying a medical image. So I have already, I hope the chest X-ray is uploaded. Let me also do that. Yep. And so now I have put the right path here, but the correct way to do is just if you move your cursor to the chest X-ray, you will see this 3 dots. Click that, go to copy path, and then you can just paste it here. I have already done that, so that's you don't have to worry about it. Now, so what I am doing here is, given that the chest X-ray image is a 2D image, it's generally stored in a PNG or a JPG format, sometimes stored in DICOM. So if it's stored in JPG or PNG format, we can use a library called OpenCV. So the way to import an OpenCV is import CV2. That's what I have done here. And the way to the code to read a PNG or JPG file using CV2 is cv2.imread, that is image read, as simple as that. And in curly brackets, just put the path, and it will spit out the image. But this is the image array. You can't just, like, if you print image CXR, it will return some numbers. So how a computer stores an image? It stores as an array or a list of list. I don't know what programming languages do you use or what programming languages you have learned. I started with C++, so I call it a list of arrays or an n-dimensional array, but like people in Python like to call it list of lists. Okay, so if you just print the image CXR, you will just see a bunch of numbers. Does not make any sense. But if you print the SAPE, it will return 629 cross 630. That means there are 629 pixels in the column and 630 in the row. And so how do you visualize these images? You use a library, another Python library called matplotlib, and the correct way to plot a chest X-ray is plt.imso, that is image so, and in that you just put the image CXR as input. Let me quickly remove this and print. You see, this looks like a very colorful chest X-ray, which is generally not the chest X-ray that we would like to see. So what's the difference? What's happening here? Any idea why this looks like a greenish bluish chest X-ray? Okay, so what happens here is If you try to plot anything using matplotlib, it by default takes into consideration that this is an RGB image. What's an RGB image? Any image that you click, somebody was clicking a photo here. So any image that you click using a camera has 3 channels, the red, green and blue. So any natural image you see, you are seeing me, you are seeing the board, you are seeing this world. It's basically consists like a combination of these 3 primary colors. But medical images are grayscale. So it's between 0 and 1. So if you want to plot something in matplotlib, you will have to use a simple argument called cmap equals gray. That means whatever you are trying to plot will be plotted as a grayscale image. And now if you rerun this cell, you will see a chest X-ray, which is generally the chest X-ray that we have seen. Any questions? No? Okay. So loading a chest X-ray, a 2D image is easy. Now let's see how to load a DICOM image or a Nifty image. So again, the same upload button which we are facing so much problem with. Have you guys downloaded the MRI? This, yeah? Okay, so upload any sequence, any except maybe this 19 KB one. Upload any of the sequence. It should also not take that time. Ideally the underscore 00001, and let's see how to upload. Yes, I'll stop. Yeah. And then let's see how to upload a Nifty file using any of the libraries that we are using. Yeah, I think they'll just check if you are able to upload or not, so I'll just wait.
Yeah, I think you have seen CT MRI folders are there. You can open that folders. There are like in
5
CT
files.
there are 4 files. Yeah.
CT there is one file.
MRI there are 4 files, I guess.
5 files. Yeah. 4
so you can open any one of those. All are DICOM. .ini.zz format. And then the same way you have uploaded the X-ray and the TGG slide, the same way.
Any questions on CV2 and matplotlib? If you are going to work in medical imaging, reading and plotting medical images is the way to go.
If you have downloaded the entire things, so you can see that like CT and MRI 2 folders are there in like any of those. Okay, you can actually. Sorry.
Yeah, if it is downloaded as a GIF, the zip folder, this Google Drive, unzip it and then load it individually.
Anyone is facing difficulty with uploading?
That is not the zip file. No, just upload it. That's not the zip file I was talking about.
Do not extract that .ini file.
Yes.
It's the format. Please upload as it is. What happened? See, if you feel like it is zip file, okay, first check the extension. If it is .ini, it's not a zip file. Or if you are completely downloading the MRI folder, it might be a zip file. So you extract it first, and then you will see the different kinds of files inside it. And then you see that like as a zip file, but they are not zip file. The extension should be .ini, and you upload as it is.
Okay, so anyone has any difficulties?
Everyone finished? Yeah.
Okay.
I think we're good to go.
So, as I was discussing, if you want to upload a nifty file, which is basically this nii.gz, you will have to use this library called nibable. So, the way to import is import nibable as nib, and again matplotlib, just to visualize what we are reading. Now, you remember like for chest X-ray, the command was cv2.imread. Here to load a nifty file, we'll have to use nib.load. But let me give you some points here. So if you upload, if you run this code nib.load and just pass the, oh by the way, same, just click this 3 dots, click copy path and paste it here. I have already pasted it so that you don't have to do it again. And if you just use this nib.load and image, this will output you something like this: class nibable.nifty1.niftyImage, data shape, affine and metadata. So when you acquire a medical image in Nifty or in DICOM format, you just don't store the image, you store an entire set of metadata. So what is this metadata? For example, let's say there is a CT scanner here at NIT Rourkela, and they are acquiring your CTs. So we need to know what is the pixel dimension. What is the pixel dimension? Some CTs or some MRs may capture the image with 1 mm thickness. Some MRI, let's say any neighboring institute, let's say some institute in Kolkata, they are acquiring image with a thickness of 1.5 millimeter. Now if you use Python, read that image, plot it, you won't be able to know what's the thickness unless and until that metadata is stored in this header file. This is called a header file. So you see here I know this MR, the slice thickness is 1 cross 1 cross 1 millimeter. That means each voxel that you will get has a thickness or has a volume of 1 millimeter cube. There can be other informations like what's the S row, what's the Y row, etc., etc. What's the affine? We'll not discuss what a nifty affine function is. We'll discuss that when we are trying to save something or download something. But for reading, you don't need the header file. Just to read the image, you don't need any information from the header file. So what we generally do is nib.load, and then I directly get the F data. So you see this was not there here. Here there was no .get F data. But here to load an FT file, we have this .get F data. So what it does is from the header, it just gets the image for us. And now if you print the image, you see a bunch of list of lists or a bunch of arrays, which means the image is here now. If you want to print the shape of the image, that means what's the volume of this image, you just have to write image dot shape and then print it. So you see the volume of this MRI is 240 cross 240 cross 155. That means the height and width of the image is 240 cross 240 and there are 155 slices. So I am just trying to plot a middle slice or any of one of the middle slices, that is the slide 60th. Again plot plt.imso image. So any idea what does this mean? Image in square brackets colon comma 60. Array slicing. So you guys might have taken the Python course. Can somebody explain a bit more in detail in the mic so that?
Yeah, so actually what it does is it does the array slicing. Here we have put the first one as colon and second one as colon, which means x and y axis it will take all the data, and the third column, the third value in that bracket we have used 60, which means it will start from I think 60 to the last.
No, so 60 to the last is again 60 colon. So I am just taking the 60th element.
Okay.
Yes. So what I am doing here is, if you have a box, just imagine the MRI as a box. I am taking the entire, like the height and width of the box, but I am just slicing it through the middle or the 60th slide. So it will just take that slice and plot it. You see what I am saying? But if you do, so what array slicing means, so if you do something like this, let's say 30 to 50, or no, I think that will not give any image. Let's say 200. Let's plot this. So you see, let me, to understand the difference, first you'll have to see the, so this is the entire MRI. You see there are many black pixels on the top and many black pixels in the bottom. But if you do something like this, 30 or let's say 40, colon 180, you see there are some black pixels cut off. So you are cutting it off from the x-axis and also the slice. So why this comes handy is, so you don't, so if you upload this image and feeding this to a machine learning image, machine learning model, the model is learning many black pixels, which you don't want. You just want to crop the image with the image that has only the MRI and then feed it to a model. So you know, this array slicing or this list of list slicing comes handy. So this is how you plot an MRI. If you want to plot any other slice, you can just change the number, and so this is, I think, plotting right across the neck. And then if you increase the number, like let's say 180, sorry, it was just 155, my bad. So let's say 120, yeah, it's trying to plot on top of my head. Okay, any questions? So similarly, you can also visualize your CT image. There is another CT image. You can try that out later, upload it. The process remains the same. You can upload it here using this upload button, change the path there and then print it. Okay. Step 2 is basic image processing. So in let's say I was talking about scanners. So there is a scanner at NIT Rourkela, let's say in building A. It's acquiring image with 1 cross 1 cross 1 thickness, and there is another CT scanner at building B, which is acquiring CT scan with the slice thickness, let's say 1.5 cross 1.5 cross 1.5. And let's say over 1 year you collected 1000 patients each or 1000 CTs each, and now you want to train a deep learning model with your own institution data. Would you combine this data and train? No, generally no, because they are of different thickness. There can be different dose because different patients have different dose labels. For example, a patient who, like, you want to see where the tumors are, you will put a high dose CT, or person who has recently gone through chemo, you don't want to add any other additional radiation in the body, you will take a low dose CT. You know, there is a dose variance, there is a slice variance. Different scanners will have their own variations, different institutions will have different scanners, the age of the scanners also matters depending on some studies. So, like, you know, multiple factors. But if you want to train an AI model with this diverse data, the model might get confused. So, what is the best approach is you do some data normalization and then feed the data. So, some very basic image normalizing step is let us say, so normalization is similar to any data normalization in statistics. Maybe when we took the statistics 101 course, how to normalize the data. Let us say we have a data x, the way to normalize it is x minus mu by max of x minus min of x. Simple, same goes for any image. You take the image, calculate the mean of the image, subtract it, and then divide it by the difference of the max and mean of the minimum of the image. And then you plot the normalized image, which does not look different in your eyes, but from a machine learning or from a deep learning perspective, the pixel values have changed. So, that is one way. You can just run this, and this will be your normalized image. Now, before the advent of deep learning models, there were some sophisticated edge detection techniques. So, how many of you have heard about Canny filter? No. Okay. So, what a Canny filter does is, so your image is a matrix, right? In mathematical terms, your image is a matrix, and if you multiply your image with another this edge detection matrix, it will plot the edges of the image. For example, you see here is a chest X-ray, so there is an edge of the lung, then there is a thorax, and then there are bones. So, can you just do some basic image processing and plot the edges? So, the CV2, the OpenCV library has a function called Canny. So, if you just pass this, you see you will plot the edges of the chest X-ray. Now, you can play around with this minimum and maximum thresholds. You can change this will change the image. You can bring this to lesser, you will see more dense edges. So, depending on what you want to do, you can change these thresholds and plot different edges for a medical image. This comes handy when, let us say, if you have a MRI and if you want to calculate the edge of an MRI for skull stripping, etc., basic pre-processing, this becomes useful there. Any questions? Nope. Okay. So, now, sorry, yes.
So, while calling the Canny method, we are passing 10 and 150. So, what was that number?
So, those are the minimum and the maximum thresholds of the Canny filter. So, there is something called Canny filter, which is a matrix, and how much. So, basically, let us say your image is somewhere between 0 and 255. Between which pixels you want to put the threshold, between which pixel range you want to put the threshold, that is basically this value.
So, those range, so pixels that will form within that range, they will be retained? Or
no, that is adaptive thresholding.
Okay.
I am not talking about thresholding, I am talking about, so thresholding is basically, okay, let me do that. Let us remove this. So, let us say gray image equals gray image greater than 150. Wait, it is still printing the edges. So, you see, this is what I was talking about as a thresholding. So, all the pixels above the value of 150 are retained, rest all is not retained. So, this can be used for lung segmentation. You see, somehow if you play around with the pixel value, you can segment the lung. But in order to detect the edges, you will have to use a filter. So, what is the basic difference here? Here above the pixel value is 1, and below that pixel value is 0. But what an edge detection algorithm does is, it takes another matrix, multiplies it in a convolution manner, and then detect the edges. and those thresholds are basically the thresholds of those filters.
Thank you.
Any more question? Okay. So, and that is actually a very good application. Let me just take some time and show another application. So, for example, image equals nib dot load. I want to load this brain MRI. Copy path. Oops, sorry. And dot get f data. plt dot mshow image, let's say 60. I don't want to plot the axis of, yep. So this is my brain, right? Now I want to just print a brain mask. So in this image, I just want to print where the brain is. So what I can do is, based on this thresholding technique, what we just discussed, you see, you can segment the brain very beautifully using very basic thresholding. Now, if you want to, so you see the brain structure. Yeah. So if I zoom in a bit, or I think this is still a poor quality image. The next step is visualization. After that, I'll discuss another interesting point. Okay. So the next step is visualization workflow. So these are all Python-based visualizations. Why do we need this? Because if our end goal is to develop some machine learning or deep learning or some analysis pipeline. You see what I'm saying? So for that, you need to upload the image in Python and then visualize it. And let's say if you ran some segmentation model, you again would like to see those images, how the segmentation happened and everything. You still need to visualize in Python. That's why Python visualization is needed for informaticians like us. But for radiologists or for clinicians who don't want to write code, there are some visualization techniques. So one of the very basic, or I would say very good visualization platform is 3D Slicer. You don't have to download it or install it in your system. You can do it later, but I have it in my system, so I can show you some basic examples how to open a 3D Slicer, load a brain MRI image, and visualize. So this is the 3D Slicer icon. And so the way to upload something is, you just, so this is my MRI folder, you all have this. I will just take any of the MRI sequence, drag and drop here. This add data into the scene window will pop up. It will show me that, okay, you want to try, you are trying to upload this sequence, and that's the volume, and I say okay, and this is the weight. Give me 1 sec. Let me close it because I already had the segmentation open, which I never uploaded. So let me try to upload it or open it once again. So there are multiple other visualization tools as well, 3D Slicer, ITK Snap, etc., etc. Feel free to use whichever you feel comfortable with. Yep. So as I was saying, just drag and drop, and this is the volume. So 3D Slicer is basically displaying the volume in 3 different views. Can anyone say what these 3 views are? Clinical terminologies. Sajai, microphone please.
Frontal, coronal and sagittal plane.
Which one is frontal?
Frontal.
So this is axial.
Sorry, axial,
This is, yes, and this is that. Yes, thank you. So if you scroll, it will scroll through the different slices of the brain, and of course you can see a big tumor here because this is a patient who had glioma. So this is a very interesting way to visualize your medical images. Now I was talking about brain mask segmentation. So brain segmentation is fine, but let's say you want to segment the different anatomies of the brain. For example, you have the cortical gray matter, basically these black regions, and then there is white matter, and then these things, this black hole like things are lateral ventricles, etc. So if you want to segment these intricate anatomies of the brain, which you know that human brain kind of of course it is very different, but the anatomies are kind of the same. The cortical gray matters will be on the side, then there will be white matter, and then there will be lateral ventricles. You can try to use some edge detection filters to detect the edges of these anatomies and segment them. Some basic, if you don't want to use deep learning, there had been methods which used edge detection for anatomy segmentation. Okay, so that's why I wanted to first visualize it and then come back to that segmentation discussion with which someone just pointed out. Now, so this is the brain MRI, and let's say I have the tumor segmentation of this particular patient. Before that, let me also visualize the other sequences. So this is the T1 contrast, post contrast, and then this is the T1 normal. You can still see the tumor here, but the difference between T1 post and T1 normal is the anatomies are much better visualized in T1 normal. I mean, the medical physicist guys will be better, will be a better person to explain this. I am not a physicist by training, but still trying to do. This is a flare sequence where the tumor, the SNFH, the SNFH signal, which is basically the extended part of the tumor, is very well visualized. And then finally, this is the T2 weighted. Yep. So these are the 4 different MRI sequences which a radiologist will or a neuroradiologist will ideally need to make a diagnosis. For example, I came there is a patient who came in with headache and something, and we want to get their MRI to check if it's a glioma or not. We'll ideally likely to have these 4 different sequences to make a correct diagnosis of the glioma. Now, if I want to also visualize the tumor segmentation on top of this sequence, so what I'll do is, okay, I think I fast forwarded a bit. So again, go to the same folder, and this is my segmentation file. I'll again drag and drop here, but this time I will not select the volume. I'll select this drop down menu, click segmentation, and then click OK. So now if I zoom in a bit, you see the segmentation is very well visualized on top of the sequence. And these are the different compartments of the tumor. So here, okay. So if you click this drop down icon and then click segmentation, you will see these 3 labels here. If you want to just look at the enhancing tumor, which is generally ideally a neuroradiologist would like to see, that is this enhancing part, you can just click this eyes just next to the segmentation icon, and if you just close those eyes, those labels will be gone, and you can just see the enhancing part. You can play around with this one, if you want to, let's say, change the opacity, you want to make it less opaque so that you want to see the background of the image, you can change the opacity, you can increase the opacity. If you want to make it a very solid label, you can change the space fill as well. So you can play around with your segmentation mask. Any question? Yes.
So are these DICOM data obtained from a single MRI?
At this what data, sorry?
So the different files that we saw in the directory, that belongs to one patient.
The
right?
same patient, yes.
So in a single MRI, we get all sort of data.
All the 4 sequence, yes.
Okay.
So that's what I said. A neuroradiologist, a patient, if comes with a case with glioma, a neuroradiologist would ideally like to have these 4 sequences to make a correct diagnosis.
And all these individual DICOM images are actually formed in the right in several layers. So how currently, today radiologists actually evaluate. They go through that those layers manually. Or?
Manually, yeah. As of now, there is definitely some AI automation going on, but yes, still now the radiologists look at those slices and manually.
So these 4 protocols that you see, T1, pre-contrast, post-contrast, T2 and flare, these are the structural protocol sequences. So there are other functional sequences, fMRI for example, functional MRI, you have PET scan. So these 4 are typically available, or these 3 rather, T1, T2, flare, they're typically available in all machines. So any place you have an MRI scanner, you will be able to get these. So the more specialized scans that are out there, you need more other kinds of machines for that. Yeah. So the reason lot of these machine learning papers that you see out there that are working with brain images, you'll see that there are these 3 or 4 protocols, is because they are the most commonly available ones. Yeah.
Okay. Any more questions? Okay. Okay. Am I audible? Like, is the mic working? Yeah. Okay. So if you don't have any other question, so that's sorry. That's all about visualization. You can try out ITK Snap. Another thing that initially I decided not to discuss, but given that today morning somebody talked about GDC portal and stuff. Okay, I'll just wait till the projector is on. Yeah. So similarly, so these are all imaging modalities, right? And if you if you are diagnosed with some sort of cancer, it's not just the imaging data, it's the histopathology data. You also have the genomic data, etc., etc. So all the different visualizations are different. So for example, this is all for radiology images. You can use something called QPath or OpenSlide for histopathology image visualization. There is a very good open source, I would not say library, but analysis platform called Xena. I can discuss. Oh yeah, it's here. Okay. So let's start with GDC. I think today morning Professor Prasanna was talking about the TCGA, the TCIA and the GDC portal. So a very quick suggestion, if you want to work with medical imaging data and specifically cancer data, this portal is from NIH, the National Institute of Health from US. You can download the data from here. You can analyze the data, format the different patients, the different information of different patient, what was the age, what was the gender, at what age this patient was diagnosed, what was the treatment given, everything is in this platform. So if you want to design a machine learning platform with multi modal data, the imaging, the genomics and the histopathology, it's readily available for multiple cancers. So you see like in the right we see the cases by major primary sites. So all the different types of cancers are here. So if you are interested in any one particular type of cancer or interested in working in pan cancer data, you can actually use this for your analysis purposes. So again, a quick glimpse about the TCIA portal. So if you just click the cancer imaging archive, so here if you go click access the data, search radiology, just taking some time to load. So here, as I think Dr. Prasanna was mentioning, if you want to design your own cohort, let's say you want to work with lung cancer patients, and lung cancer not only the primary one, by primary I mean patients who developed only lung cancer as their primary cancer, but also want to work with lung cancer which are metastasized. For example, somebody had brain cancer that got treated, but it metastasized to lung. So you can actually design or build your cohort here. I'll give you a very brief, go through this TCIA portal. It's just loading. Let's see. Meanwhile, we will not be, so the talk is all about medical imaging data, but if you're still interested to work with, as I was mentioning, the genomic data, you can go through this Xena browser. So this is a portal by UCSC, and here it's very interesting. If you are working with genomic data, you can visualize all set of, you can get access to all the different data sets, the breast cancer, all the GDC data sets are also normalized here. You also have access to the pan cancer data, etc. You can quickly play around. You can do multiple things with this portal. Like I can give a small example maybe. So for example, I am interested in brain cancers mostly. So if I go to the GDC, TCGA, glioblastoma, which is one of the most aggressive form of brain tumor. If I go here, you can get all the copy number data, the gene label, the DNA methylation. Somebody was talking about DNA methylation here. The RNA sequencing data and the different phenotypes, etc., etc. So if you click visualization here, and so this is very important. Like if you just want to look at which genes are expressed in which patients, so select a study to explore. Just click here, GBM, which is glioblastoma, the TCGA GBM. Then select your first variable. First variable. Okay. Now I want to see what is the genomic, the structure. So I know which gene I want to see across these patients. For example, for glioblastoma, the most interesting gene that you would like to see is the IDH1. So I just clicked IDH1, and here I can go to advanced, or let me just keep basic and see the gene expression data. See, okay. And then again I want to see genomic between KI67 or any other, let's say TP53, somatic mutation, done. So it's loading. Yeah, so you see this is the entire genomic distribution of the TP53 and IDH1. So you can see papers who actually derive their hypothesis from these set of genes and then do analysis on their own data. So just to have this in mind. Coming back to TCIA, it's still loading, so I'll just keep it there. We'll come back to TCIA again. Okay. Any questions regarding visualization? No? So that means two things: either you understood everything, or you did not understand anything. Okay. So now basics of pathology informatics. So I hope the TCGA, that SVS file, is uploaded. So in order to visualize the histopathology image, okay, brief introduction about the SVS file. So this SVS file actually contains a histopathology image. Any idea what an histopathology image is? I think we briefly discussed in the lecture, but again reiterating how an histopathology image is acquired. So first step, if we suspect, or if the radiologist or if the oncologist suspect that you have a cancer or if you have a tumor, they'll first do the imaging. Let's say I suspect that somebody is having glioblastoma. I'll first do the MRI. I can see the tumor there, But very important, just detecting they have brain tumor is not enough for treatment. So we need to know what is the subtype of the brain tumor. So if it in glioma, there are 2 or 3 main subtype. So just now we saw IDH1. So if the IDH1 mutation is there, that is, any idea what, if a patient has IDH1 mutation, or if it has wild type, what does that mean? Okay, I'm assuming you guys are not from genomics background, but I think if it is IDH1 wild type, it's glioblastoma, and if it's IDH1 mutant, it's either oligodendroglioma or astrocytoma. But how do you came to know about this IDH1 mutation? The imaging can't say that. So what you do is you put a needle through the skull, or there are different techniques, but you do a needle biopsy or an open skull biopsy depending on the patient, put a needle, get some tumor sample out, slice them, put it under a scanner and get the histopathology slide. So that is basically for one patient in this SVS file, the histopathology slide. And then I was talking to someone in the morning, the next step after getting the histopathology, the staining and everything that we discussed in the lecture, they take the slide and then they get the RNA sequencing or the DNA sequencing. So basically they get the genomic data from each cell. And that is how you determine if the patient is IDH1 mutant or wild type, or it has TP53 mutation or not, or different types of genomic studies. So the second step is the whole slide image, and the third step is the genomics. Combined all, the oncologist decides what is going to be the treatment. So to visualize a whole slide image, you need to install this tool called OpenSlide, and Python also has an OpenSlide Python, and then Matplotlib and Pillow. So I'll just run it, it might take some time. I hope you will not get any error in this Colab files, because I have checked it and it's kind of error proof unless and until you have messed up with some installations. Then the step 2 is load the SVS image. So again I have copied the path, and yep. So you see there are 4 levels of this image. What does this mean? So a histopathology image is it's not like an MRI, which is just like one magnification. So histopathology image has different levels of magnification. So it has either 5x, 10x, 20x, or depending if you have a very costly scanner, 40x magnification. So in 40x magnification you can see each cell and the distribution of each cell. 20x magnification you can just see something like this, which, well, I can see the tumor here, but I don't, if I want to know the tumor microenvironment, I would like to look at the 40x magnification. Okay, and the slide dimension is huge. So MRIs we were dealing with 240 cross 240 cross 155, but histopathology it's like, you see, I don't know, 1,25,661 and 61,000 something. This is the size, this is the number of pixels of the histopathology image. So it's huge. And that's why the loading and uploading and downloading was taking time. Okay, so I'll just discuss a very basic visualization. You just use the open slide, that is slide.getThumbnail. I just want, so basically I'm compressing this entire big 1,25,000 cross 61,000 to 1,024 cross 1,024. So I'm just downsizing it so that we can visualize. So this patient might just have 2 different slices there. So these are the 2 different slices. You can read, adjust the heights and visualize it. And if you want to look at, so here I'm just visualizing the level 0, but if you want to visualize different levels, you can just change this level number and look at different magnifications. Okay, so I would just like to summarize the first step of the basics of medical imaging. There are a few public medical data sets. One is definitely TCIA and TCGA and GDC. But if you want to access other data sets, for example, for brain tumors, you would ideally want to access this. Have you guys heard about the Brat's Lighthouse challenge? Brat's Lighthouse. So Brat's Lighthouse 2025 has multiple challenges, It's for brain tumor segmentation or glioma segmentation. Yeah, so there are 11 different tasks: pre and post treatment, adult glioma segmentation, pre-treatment intracranial meningioma. So one primary type of brain tumor is glioma, another type of brain tumor is meningioma, and then pre-radiotherapy, pre and post metastasis, etc. So if you want to train, so where I'm going is, if you want to train a deep learning based segmentation model for tumor segmentation, a good start is the BRADS dataset. Download this. It's somewhere in the range of 30 to 40 GB, which I don't think it's that bad. Download it, visualize the images, and try to run a deep learning model. So these are few data sets which comes very handy if you want to train some basic things to try out as a start, and then from there on you can move on to much denser deep learning projects. I'll try to share a Google Doc in the last maybe in the last day, where you have access to all this, or where you will have the links to access to all these data sets and few basic codes that you can use for pre-processing. So any questions on the basics of medical imaging? Sorry? Question, yeah.
It's a question related to the GDC portal actually. Do you generally use GDC transfer tool and the manifest thing? You add to the card, manifest, and then GDC transfer direct?
Yes.
But there are some images which are like 2 GB, 3 GB, and there are thousands of images.
Yes. Oh, so you don't want to download them in your local computer.
Okay.
You should ideally download them to your server, which has TBs of space. I mean, but the good part is, like, if you know that these are the 10 different patients you want to download, then you can just add the 10 patients, and yes, they will again be 30 to 40 GBs in total. But I mean, SVS files are large, so you can't help with it.
Any questions?
No questions?
Anyone?
Okay, should we take a break before pyradiomics, or?
Yeah, so we'll take a break shortly, just waiting for tea to arrive.
Okay. Okay, then I'll just start with the introduction of pyradiomics before taking the break. Okay, so till now, what we discussed is visualizing the image, what are the different imaging modalities are, and how the segmentation, mask, and all these things looks like. But, you know, deep learning models generally take the entire images input, do some, okay. So summary is, pyradiomics is an important tool for medical image visualization and generating features. We'll take a break.
So we'll take a break, so 20 minutes is fine, 15-20 minutes, yeah? Okay, so we'll reconvene after 15-20 minutes. In the meantime, if you have not been able to download any specific data set, then you can start your download and go also. If you need any help, let us know.
So also, I want to let you know that, I mean, I know that it's new for a lot of you. You're new to coding. All these lectures are recorded, so you can go back and watch this at your own pace, and then you can pause and then try to download videos, look at what portions of the code he's editing, and then try to do that. Okay, so you don't have, in case you're not able to follow along at the pace, don't be worried. You have everything that's recorded. It's on YouTube, so you can go back and watch everything again.
And if you have any questions, feel free to reach out to me anytime till 30th. Okay. Um, okay. So we'll start. We'll reconvene. So any questions from the previous sessions? No. Okay. So now what we'll try to do is same. Go to the folder and try to download this underscore radiomix IPython notebook and upload it in Google Colab, similar to what you did to the for the underscore basics file. So let us know when you have already uploaded the underscore radiomix file. We can start. No, but
again actually we have to do the same thing. Same thing actually you have to do. You have to first download the file and then you have to upload. There is a separate section called dataset. There you have to upload the data file. We already told you, right?
And for the dataset part, just again click this arrow and just upload the brats glee the all zeros underscore 0000 and this 000 file. So like let me view as a list. So there are 5 different files in the MRI folder. The patient name, but followed by an underscore 4 zeros underscore 0001, 0002, 0003 and no underscore, nothing, just .nii.gz. So you just have to upload the first image and this one without any underscore 1. Just upload these two nifty files in. Okay, let me try to make the screen a bit bigger. Oh, white background, I have no idea how to do in okay. How about now? Is it visible now? Okay. Wait, yeah, finally I figured out. Okay, so you'll have to just upload the first and the last file. So let me know when everyone has at least done this step and then I can start.
If you are facing any problem, just raise your hand.
Yes.
Any problem, if you are facing.
And please let us know, we can wait because this is a very critical step. Because as of now, we just discussed how to visualize medical imaging. Here actually we are extracting some features which can be used for downstream machine learning and deep learning applications. So this step is very important. Yeah, so just the first and the last. Yeah. You guys have uploaded? About you?
Yeah, actually we can proceed. I
No,
think
we'll
there is no
just wait
problem.
for the guys who just joined now.
Yeah, okay, okay.
So I'll reiterate it once again for the guys who joined just now. So in the folder, you will have another notebook named NITR underscore session 1 underscore radiomix. So again, upload it. So upload it to Google Colab, and upload. You might also have this MRI folder. Just upload the first and the last file. Similar to the previous exercise, click this upload icon. Just upload the first and the last file. So first step is, just repeating for you, first step is upload this Python notebook which is there in the folder. Upload this in Google Colab, similar, file, upload notebook, or right click, open Google Colab notebook. Are you able to upload? And then click this upload sign. You might also have an MRI folder. Upload the first and the last file. Okay. Okay. So in this exercise, we are going to extract something which we call the radiomix features. So why the term radiomix? So anything, any thing that has something to do with medical features, it's called omics. And if it's extracted from radiology images, MRI, CT, chest X-ray, it's called radiomix. Similarly, if you are trying to extract something from histopathology images, you can call them pathomics. And if you are extracting something from gene data, you will call it genomics. And if you are working with all of these data combined and trying to do some analysis, you can call it multi-omics model or multi-omics data. Okay, and so radiomics had been very popular. Actually, Dr. Prasanna did a lot of work in radiomics. Basically, he was one of the starting person who did radiomics and a lot of work. And when radiomics got famous, Harvard group, there is a group in Harvard, AIM, AI in Medicine. They made a library public, or they made a Python library called PyRadiomics. So if you have the radiology images, and the tumor segmentation masks, you can very easily extract these pyradiomic features. So what are radiomic features? So radiomic features are, okay, I'm sorry, I'll just change the theme to light. Okay. Is it visible now? Yep. So what are these radiomic features? So they can be shape based features like what's the maximum diameter, what's the diameter, the perpendicular diameters, what's the volume, what's the shape, etc., etc. And then there are shape based features, and then there are intensity based features like the GLCM, the gray label co-occurrence matrices, and different radiomic filters like Hog, log, Laplacian of Gaussian, etc., etc. So the idea of extracting these features is something like this, that to study, so for example, if you have a solid tumor structure, so what are solid tumors? So any tumor that has some shape are called solid tumors. For example, glioblastoma, we saw that it's a solid structure. But again, the tumor in blood, which is just in the blood, there is no solid structure, it's not a solid tumor. So for any solid tumor, you will have a shape. The idea is, can you extract some shape and intensity and texture based features from these solid tumors and make a prediction. So what kind of prediction? Well, the first thing that comes to my mind is, if this is the structure of the tumor, will this patient respond to a certain kind of treatment or not, right? Or if it's given treatment or not given any treatment, how long this patient will survive. So that's called the overall survival analysis. Will this tumor after treatment will recur or not? Multiple types of analysis that you can do. So that's the idea about like why we want to extract these radiomic features. Of course, later, I mean now as we speak, there are much sophisticated deep learning features which can do a much better job, much stable jobs, like much stable predictions. But yes, I mean before deep learning features, pyridiomic features were very important. It's still important, people do use them, but yeah. So what do we need for extracting radiomic features? We need the medical imaging data, we need the segmentation mask, and then finally we'll extract the features and then do some machine learning or statistics. Now the first step is install pyradiomics. So you can directly install it from the AIM Harvard GitHub repository, or you can also use something called pip install pyradiomics. Don't do it, because now I think they have an issue with pip install pyradiomics because of the NumPy version. So as of now, don't use pyradiomics. If you are trying to extract radiomic features in your local environment, by local environment I mean similarly install Anaconda, make a Python or a Conda environment and then install pyradiomics there, you can manually change the NumPy version, or just very simple, use this pip install git https blah 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 pyradiomics.git. So again similarly run this. By the way, so these are not the only set of radiomic features that people use. You can use these features for a start, but if you are trying to develop a machine learning model based on your data, or if the problem statement is different, you can try to think, that's a good research problem, you can try to think about your own radiomic features, like what other features you can calculate from whatever segmentation masks you have that will make a better prediction. For example, I can think of one of our research idea which deals with not solid tumors but vessels of the lung. So lung vessels, of course there is a shape, there is structure, intensity, everything, but in order to do some meaningful prediction, vessels are very irregular in structure. So the first thing that comes to my mind, okay, how long is this vessel? How many branches this vessel has? What's the tortuosity? That means what's the curvedness, what the tortuousness of these vessels are. So these are new type of radiomic features that you can think of if you have a different problem statement. So like from solid tumors to vessels, I thought of this set of features. If you have something else, you can think of some other set of features. Now given that I hope everybody has this the two files, the _0000 and the nifty file uploaded. So I'll tell you what these files are. I think I already visualized. So the _0000, that file is the T1 post contrast sequence, and without any underscore, that's the segmentation file. So what I am doing here is I am reading both of them using another new library called SITK. You can use nifty as well. I just use SITK because I just wanted to introduce another library. So you can use nifty and SITK. Like basically I am trying to do two things at the same time, another library and pyradiomics. Okay. And before Nibble, actually we used to use SITK. That was the only or the most prevalent library for uploading DICOM and nifty files. Okay, so the command goes something like this: sitk.read image, and then you read this image, that is basically the T1 post contrast sequence, and then sitk.read image, the segmentation file, that becomes our mask. So let me quickly try to import matplotlib as blt. Oops, sorry. blt.imshow 1 second. Anyway, I don't know what's happening. Let me try to rerun again. There is some error. Okay, the image size is 240 cross 240 cross 155. So we'll see how to extract the radiomic features. So from radiomics, import feature extractor, and then from feature extractor you call this function radiomic feature extractor, and then what you do is you try to binarize the mask. That means these are the different compartments of the tumor. The green is the necrotic core, and then the brownish one is the enhancing tumor, and then this yellow is the PE, which is basically edema. So now what ideally you do is pyradiomics does not take this multi color or multi label input. So you will have to binarize this. That means anything that is not 0 is your tumor mask. So if I were to visualize it, let me try it again. Let me try to read that in Nifty. Image is equal to nib. Let me just try to copy this. Okay, it works. So this is the MRI sequence, and let me try to plot the mask. Let me see. So you see, you can see the 3 different labels of different grayscales. Now what I'm trying to do is you see what happened? From multi-label mask, if I just do anything greater than 0, that means anything that has labels becomes 1. So this is a whole tumor mask, not just compartment wise, and you want to extract the information from each of this from the whole tumor now. So that's what I did here, and then if you just run features is equals extractor.execute and pass the image and mask, it will output this pyradiomic features for you. And you see these has all the different features like shape, elongation, flatness, major axis length, maximum 2D diameter, blah blah blah. This has first order statistical values like kurtosis, maximum, mean absolute deviation, mean, etc., etc. And then intensity features like the gray label, co-occurrence matrix, difference variations, the IDM, IDMC1, joint entropy, etc. So these are all the different set of pyradiomic features that you can extract. So this is the feature name, and this is the feature value for the whole tumor. Now it's a Python question, nothing to do with medical imaging. So you want to study, as I said in the session before the break, that neuroradiologists, of course, all the tumor, like all these different compartments are important, but neuroradiologists are particularly interested in the enhancing tumor. So how would you just take this particular level, which is segment 3, and extract the radiomic features? So what will you change here? So as of now we did mask equals mask greater than 0. So that means anything that has 1, 2 or 3 value becomes 1, and it looks like this. This is the whole tumor. So just to plot one segmentation label, what should we do? Answer. Let's say the value is 1 something. Mask not equals 1. Greater than 1 means it will include 2 and 3 as well. Wait. Okay, let's see first like what are the unique values. Oh, so this I see what's happening. Let's see. Okay, so there is 0, 1, 2, 3. Now if I just want to plot 2, so you see this is the last part. Now if I want to just plot 1, I'll have to rerun it again. It's a bit bad coding on my behalf. So this is the necrotic core, and if I want to print 3, which is basically the segment 3 we already saw. Yep. So this is the enhancing tumor that we want to extract the radiomic features. So I will just rerun this code and wait, what happened? Oh, sorry, my bad. I see. Okay. So when we are doing mask equals mask greater 0, it prints, wait. Oh, I see. We are changing the image to nifty. My bad. Mask equals 3. Now it should run. Yep. Yep. So now you see this is the radiomic features of that enhancing tumor region. Oh yeah. So what happens is this PyRadiomics library only takes SITK image as input. So the moment I converted it to nifty, it was showing as an error. So what I just did is, rather than, so initially let me comment out the code here, mask equals mask greater than 0. So if we want to calculate the radiomic feature of the entire whole tumor, we are just doing anything greater than 0, that is 1, 2 and 3 becomes the mask. But now if we want to calculate the radiomic features of only that particular region that we segmented, that we just saw, you just do mask equals 3. So similarly, if you want to calculate the region, the radiomic feature of another region, just do mask equals 1 or equals 2. So this is how you calculate the radiomic features either of the whole tumor, all of or different segmentation labels of the tumor. Any questions? Yes.
Yeah. How did we create the segmentation mask DICOM image?
So you are asking like how did we get this?
Yeah.
So the dataset had the segmentation with us, like they provided the segmentation labels. How that was done? It was done in a semi-automated fashion. What is semi-automated? So you train a deep learning model with the MRI scans and the tumor segmentations, and let's say you now have 100 MRI scans from NIT Rourkela, let's say, and then you run the deep learning model, or you run the inference of these deep learning models on those images. You get the tumor segmentations. They will have some errors. Send it to a radiologist. the radiologist will manually verify that these voxels are wrong, these voxels should be there, these voxels should not be there, and then finally they will save the segmentations after the correction, and then you can use them to extract the radiomic features. As of now, for this data set, the data set providers gave us the segmentation labels.
Got it.
Yeah. So that's what I said. If you want to work on your own deep learning based tumor segmentation model, not only for glioma but for meningioma, metastasis, all the different types of brain tumors, you can actually take a look at the Bratz Lighthouse 2025 challenge. So that like has almost it's a state of the art data set now. Okay. Any more questions? So are we clear with extracting pi radiomics, because this is very critical. Like if you understand this notebook, and if you have the right set of data, and if you know how to do some statistical analysis after getting the output, you are as good as to infer your own outcomes and write a paper maybe with just these set of features. People do that. So this file is very important. You can, yeah, sorry. Okay, okay, let me write it here.
Guys, actually you have to do the same thing, okay? Just don't sit. Just do the same thing, whatever things right now is going on the screen. Actually, you have to do the same thing.
But I will share with you these files again, like with all the modifications and stuff. So yeah, but still keep like.
But still, actually, you have
practice.
To do, because for practice also you have to do.
Yeah, you don't have to feel left out. But I'll it's good to get your hands dirty by just doing basic things like visualization. It's simple, right? And the best part is in 2025, if you just write image, Colab will write the entire code for you. You just have to do tab, tab, tab. If you like it, it's fine. Otherwise, just write your own code. Just start writing few lines, it will write the entire line for you. So you guys should feel as confident as a coder now, because like when there was no Colab, nothing, people had to manually code. And if there was one error, you will figure out after 10 steps. Here, Colab is by default writing the code for you. So if you know the concepts well, coding is not a problem now. So the same course previously, 2-3 years back, we used to teach from the very basics of coding. But right now, with Colab and everything is like so integrated with AI. So if you are writing a code in Colab, it's integrated with Gemini, because Colab is from Google and Gemini is created by Google. If you are writing code in, let's say, I use VS Studio. Sorry.
I think one thing are you guys also facing some problem, right? Like not imported something. Like there is a package or a library called Radiomix that actually you have to first install.
Oh.
Okay. So just type pip install and that name.
No. Use this. Are you guys able to install this?
I think you guys install, right? Those things.
Installed, right?
Like some of them are facing same problem.
Oh yeah. Start with this one. Don't use this pip install PyRadiomix because it has some bug. Use this one. You guys use the PyRadiomix, right? Able to extract radiomic features, right? Okay. I was in shock that I skipped the installing radiomics part and moved to. Okay, so the visualization code is this. Are you able to? Okay, let me try to zoom in a bit so that you can clearly see. Is it visible now? Okay. So this Bratt's glioma, all this images, it's just for one patient, but if you want to download the entire cohort, you can download it from the Bratt's Lighthouse challenge. This is a patient, this is basically the first patient from the Bratt's glioma pre-treatment tumor segmentation challenge. So many TCIA data sets have the radiology images, either MRI or CT, and the tumor segmentations available. So if you can download them, pre-process them, well, pre-processing can be a bit tricky. It's not difficult. If you can pre-process them very well and use these 10-15 lines of code that I shared with you, extract radiomic features, do some very basic statistics, I think that will be a very good analysis. And if you have a good outcome, like you can clearly see that there are difference in patient features, etc., that will be a very good finding for your maybe class projects or master projects or something like that. So are we done with the image visualization? Anyone who is not able to use this? Mm-hmm. That is here. So this is for the whole tumor. Let me just comment it. But if you want to print a particular region, like this is the ET, enhancing tumor, you can just use this. I'll just comment this out. And if you are facing any problem with the coding, just rerun the files from the top, because I have a bad habit of naming the same. So for example, here I am changing the mask label, but I am renaming it as mask. So you might get some error, because I have this thing like I don't want to name it as mask 1, mask 2, mask 3. So after some iterations I will forget what was mask 1, what was mask 2, and I will end up with mask 31 or 32. So I just keep the naming convention same. Okay, so everyone done. Everyone can extract paradigmics features. Great. Okay. So what we'll do is, for the next 20 minutes, I will briefly touch foundation models, and I think Professor Prasanna will discuss foundation models in detail tomorrow. And okay, so what are foundation models? So like if you, okay, any questions before we move ahead? No, right? Okay, yeah, please.
There are two images. So first I upload 300, and then मतलब sequence is wrong. We first upload downside image, first upload, and second.
Yes, that is a problem.
This also
because mask, because this image, you see, this image is the mask.
Yes.
Of that particular patient, and this image is the image. So that will be a problem. Great that you asked that question. I think another question would be, is it a problem if we, rather than using 0000, if we use 0001? which is basically like there are 4 different sequences here. Underscore 0000 is the T1 post contrast, 0001 is the T1 normal, 0002 is the T2 flare, and 0003 is the T2 weighted. So just for this radiomics experimentation, I extracted the features for T1 post contrast, but the features if you upload 0001 will be different, and the features if you upload 0002 will again be different, and 0003 again will be different. So actually tomorrow I will discuss, so same patient, different MRI sequences, but the same tumor mask, different features. How, and let's say different features means different analysis. So how will you combine these and make a prediction? So that we will discuss tomorrow. As of now, just extracting radiomic features. Okay, so foundation models. So anyone heard the term foundation models? Foundation models, like if you, I don't know if you guys use LinkedIn or not, if you open LinkedIn and if you follow some groups, all the papers or most of the papers are on designing foundation models, fine tuning foundation models, training foundation models, blah blah blah. So foundation model is a new term. It's basically a very large deep learning network trained on like a lot of data. For example, ChatGPT, I'm pretty sure everyone have heard about ChatGPT, Gemini, etc., etc. ChatGPT is a foundation model. What is that? It's a model trained with the entire data that OpenAI has, which no one knows what is that. So if you have large amount of medical imaging data and train a deep learning model in a self-supervised manner or a supervised manner, if you have the labeled data, that people are calling nowadays as foundation models. So foundation models seem to be improving. So for example, a model trained on, let's say, just 1000 chest X-rays versus a model trained on 20,000 chest X-rays, 50,000 MRI and 10,000 CT scans, everything combined, is performing much better than just a model trained with 10,000 chest X-ray scans. So foundation models have proved to be very successful in downstream tasks like anything, thoracic disease, like pneumonia classification, tuberculosis classification, tumor subtyping, etc., etc., outcome prediction, etc. So why foundation model is important in radiology? Foundation model is definitely important in the field of AI, but why in radiology? Well, medical data sets are small and expensive. So for example, if you want to train a normal, like a deep learning model, non-medical deep learning model with just car images, you will find like millions of data with just car images. But will you find millions of brain cancer data set or a brain cancer MRI scans? Absolutely not. So in case of medical imaging, training a foundation model becomes important. So you might have 20,000 MRI scans, like, and let's say 70,000 CT chest X-rays and like 100,000 CT scans. So combined, this becomes a big enough data set to train a model, which is not possible individually. And another important thing is like medical data is very complex. So if you see like natural images, there is a car, it will be just a car standing on a road, and I am hoping all the cars will stand on a road. It will be very funny if a car is on water. So most of the cars will be on road. But medical data is very complex. It has different anatomies, different diseases. For example, brain can have Alzheimer's, brain MRI can have tumors, brain MRI can have hemorrhages, CT scans can be taken for pneumonia, CT scan can be taken in the same region for cancer. So medical data is very complex. So training a foundation model for medical imaging data is also very different. And there are very few labeled samples. So if you ask a radiologist here, we have 1 million data annotated, they'll just be very mad. We don't want. And then faster convergence and more stable training. That's of course like the only critical thing in medical imaging is we need best accuracy. We can't compromise with accuracy. You have to be 100% accurate. I mean, almost near 100% accurate at this, always, because like you can't say, well, this patient will die with a probability of 60%. That's very bad. you'll have to be very accurate. Okay, so as of now, the Google Colab files that we used, we did not use GPU, but now to even run these foundation models, we need GPUs. So how to turn on a GPU in a Google Colab notebook? So I'm going very slow here, just follow the steps. Click this runtime thing. Oh. Okay, so click this runtime. I hope everybody can find the runtime. Then go to this change runtime type. Okay, click that. Here I am hoping your option will be like the CPU. Change it to the T4 GPU. Is it already there? Because I sent you the IPython notebook, so but still it's better to be sure that everybody has the GPU, and then save. Okay, so we all have GPUs now. So like we are calling foundation like this, deep learning models trained with large data foundation models now, but like they were always there, like since 2019 maybe. So NIH, National Institute of Health in US, has made a chest X-ray dataset public, which has 14 disease labels, and I think they have almost 130K data, maybe more, I don't remember exactly. And then they have trained a DenseNet. So do you guys know what a DenseNet is? DenseNet? No? No? Okay, then let me try to give a very brief basic about what a deep learning neural network is. So like how to explain? So the first neural network, I mean we all hear the term AI now, but AI in some different form was there in 1946. In Cornell University, there was a lab, Cornell Aeronautical Lab, where Professor Rosenblatt discovered a one-layer neural network, which is now called the Rosenblatt's perceptron. It was just one layer, and you feed in something, it will output a data. So he basically used that for I think character recognition. So he gave handwritten 7, handwritten 8, 1, and that predicted that, well, this is a the text which is handwritten is now 1. So that was the first neural network. It's now called Rosenblatt's perceptron. And then there were multiple models. I would say the first deep neural network was AlexNet. I mean maybe there were a few models before that, but AlexNet became very famous. And then following AlexNet, the big boom happened in AI when ImageNet dataset was made public. So ImageNet dataset is still very famous. It has a lot of images, and that basically favored the way of deep learning training. And that is where the convolutional neural network started to enter the domain. So what is convolution? You have an image which is like a 2 cross or like let's say height cross width, like a 240 cross 240 image. You take a convolutional filter, which is again a matrix, and multiply that for the entire image and then encode the features. So that is a convolutional neural network. AlexNet was a convolutional neural network. Now what happened in the year 2016, I just started undergrad, and this paper came in. The paper name was, basically the model name was ResNet. So ResNet, I mean till that time convolutional, yeah, it's perfect, it's working accurately, blah blah blah. But ResNet, what it did was, I can just show you the ResNet structure. So this was this is a figure from the ResNet paper. So this is the convolutional layer. There is another convolutional layer. X is the input. If you just remove this identity part, that was AlexNet. But what ResNet did is it just added a residual network. So the output, rather than becoming f of x, which is a function of x, and function is basically the layers, it becomes x plus f of x, and that changed the domain. ResNet, or residual network, a network with residual connection, was able to perform very accurately on multiple different tasks. So that was one change that happened in AI. And then following that, in the year 2017, another paper came, which is called DenseNet. So what it did is, I'll just show you the structure. Yeah. So they were like, okay, why will we add one residual connection? Can you wait? Yep. Can you see this image? I don't know. Yeah. Why will we add one residual connection? We will add multiple residual connections. So this becomes a dense network, like electric wires. If you see, I don't know if it's there now, but like, in my village there is one pole, multiple electric wires coming, and no one knows where it's going. So dense net, and that outperformed ResNet. So for a long time, DenseNet was one of the state-of-the-art method. By state-of-the-art method is it outperformed all the other existing methods for multiple different tasks. So coming back, so DenseNet was trained on this NIH chest X-ray 14 dataset, and it was made public by Torch Vision, which is a model by PyTorch. So you don't have to train anything now. You can just download the weights and use this for your inference. So now, for example, you ran this code and you have this model. Now let's say you have an image, you got an image from the neighboring hospital, and you want to see what's your this pre-trained DenseNet performance on this particular image. So I'm just randomly creating an image. This is a random function, and then if I run this, wait. I'll have to print the output as well. So this is the output. This is the output feature from the foundation model. So what to do with this, we'll discuss next day. But this is how you download a foundation model. If you have a chest X-ray, extract the features, and then from there on you can extract multiple different downstream tasks. You can do multiple different downstream tasks. But the objective of this session is, this is how you download a foundation model. You don't have to train one. You can just download existing foundation models. Similarly, there was another function or another library called the Torch X-ray Vision. They trained on, so not only the NIH 14, the chest X-ray 14 dataset, but also other big chest X-ray datasets. The CheckSpurt, the Mimic CXR, the PAD chest, these are all like big datasets in itself. So overall, this become a huge dataset, and they again trained DenseNet on this, all of these datasets combined. And you can again download this very easily in Google Colab. So what I'm trying to show is you can download all these foundation models here. Then so these are already there in these GitHub repositories for chest X-ray Vision or Torch Vision. But now if you want to install something from Hugging Face, so this will throw an error, hopefully. So how many of you have heard about Hugging Face? Have heard about Hugging Face? Okay. So why do you think this error came? So there is an error here, right? So why do you think when I'm trying to download this model BioVIL from Microsoft, it throws an error like this? Just read the error, I think you will.
I think we haven't added our token, Hugging Face token, we haven't added to this.
Exactly. So what you'll have to do is, you'll have to create an account in Hugging Face. So Hugging Face is basically the name Hugging Face, the smiley. So you create an account, I already have one. You go to settings. Oh, I'm being fast, sorry. You don't have to do it now, but just like take a look. You can figure it out on your own. Just create a account in Hugging Face, go to settings, and then here you will get your access tokens. I don't remember my password. Anyway, so from here you can get your Hugging Face tokens, and then if you upload it here, it will allow you to download the foundation models. If you are really interested in using these foundation models, I can find out what my password is, come tomorrow and then download this. But anyway, similar for medclip. Medclip demo I'll show tomorrow because I think we are running short of time. Medclip and medsam. So SAM, how many of you have heard about SAM? SAM was the segment anything model made public by Facebook, and that was adapted for medical image segmentation. No? Okay, so tomorrow we will discuss medsam. It became very famous overnight because if you send a medical image, something like this, and if you just like put a bounding box, it was able to segment tumors really accurately. Just imagine, the radiologists now don't have to segment pixel by pixel. They just draw a bounding box like this, and the segmentations are there, and you can extract radiomic features and do so many things. So this became very, very famous overnight. Okay, so the final thing that I will discuss today, because somebody mentioned Monai, I think you mentioned Monai. So yes, so Monai is a very I would say very user friendly tool to either input medical image or train models using medical imaging, because Monai has made all the models, for example SWIN, VIT, I'll discuss that later, but all the models that you need public. So you don't have to write code, just import them and run your own foundation model. So for bioinformaticians or biomedical informatics students like you don't have to write the code explicitly now. Your task is to run the inference and work on the results mostly. So if you are using Monai, that basically simplifies your life. You don't have to deal with much back-end coding. So simple Monai installation is very simple. Pip install Monai, Nibable and INOPS. INOPS is basically Einstein operations, which are basically just simple matrix multiplications and stuff made faster. So if you run that, these libraries will be imported, and then as I was saying, for example, if you want to write the code of SWIN unitar, 500 lines, but if you just use monai.networks.net import SWIN unitar, 1 line. So that makes your life very easy. And then some basic pre-processing like intensity scaling. We discussed intensity, why we need to scale the intensity, and resizing. So I will load. You don't have to do this because this you can try out in the in your own time. It's fairly simple. The CT. So I uploaded the CT image that I had. We'll use this CT image again tomorrow, so don't delete these files, please. Okay, it's still uploading. So yeah, so you see what I was saying, like Monai has already trained the SWIN unitar with a large data set and has made the weights public to you, and you can basically just download the weights and start using these foundation models, which we'll see now. So my image is loaded. My image shape is 302 cross 302 cross 276. Now I will do few basic normalization steps, that is the intensity normalization. So somebody told me about the Hounsfield unit, right? I think you? No. So basically you described how the C2 is acquired and different organs have different absorption, right? So in CT, exactly, I wanted to bring that here. So basically different organs will have different Hounsfield units. For example, if you see in a CT, the bones will be very white and shiny, whereas you can't even see the fluids, the bloods, unless and until it's contrast enhanced. And the soft tissues and the organs will have different images. You have pixel values. In CTs, you have Hounsfield units. So for example, here you took a very high dose CT, so the Hounsfield unit will be very, or the intensity will be very different, and a low dose CT will have intensity of a very different scale. Again, to train a foundation model or even get the inference, the features, you need to normalize it. So what I am doing here is from minus 1000 to 400, I am normalizing it between 0 and 1. There is no information loss here, it's just like I am normalizing it. I am not cropping out all the other values. So anything that is below minus 1000 and above 400, I am not deleting them, I am just normalizing it. So it's not information loss. and then I'm resizing it. Again, like here my image size is 302 cross 302. There will be another image which will be 512 cross 512. Deep learning models can't handle this, like, you know, different image sizes. So the best idea is to just resize everything to 96 cross 96 cross 96. Yes, here there is information loss, no doubt, but that you will have to, you know, deal with. So now my final image is 96 cross 96 cross 96, and then I download the weights. So these weights Monai has already provided us, so it's very easy. And then I declare, or I initiate the model here, upload the weights in this. So torch.load basically uploads the load in the the weights in the model, and this is basically the entire model structure which you don't need to know what a Swanian unitary is as of now. And then you just feed in the CT here, and it outputs a feature of say 1 cross 768 cross 3 cross 3 cross 3. So what to do with this shape we will discuss tomorrow, but today I just briefly discussed these are all the foundation models that you would like to use. These are all the features you can extract. It's very easy. Foundation model is not something that you can't use. It's very easy to use. You see just 2-3 lines of code and you can do multiple downstream tasks using this. Any questions? If not, then I'll, yes.
I have one question. So
yeah.
I think the image that is the image path that is given in this particular collab is not with the Google Drive.
So you are talking about this one?
Yes.
Why not? Like I just uploaded this.
Okay, I think I'll use the CT folder. I think
Oh,
I
yeah,
didn't
it's
look
okay.
at that.
Anything else? Okay.
So this course, this code has been shared with the participants?
Yes, all these codes are shared.
In that group?
In that.
Please share in that group itself, collab.
So all the additional things that I am discussing, I'll compile as another notebook and I'll share it to you. And as I said, all the data sets that I am mentioning here and there, I'll again send a link so that, you know, after later if you want to work with them, you can. Okay, thank you.